Now we are opening the second session of drug delivery therapeutics. In particular, we are going to discuss about the current progress and the state-of-the-art technologies in drug development of the drug delivery carrier and associated clinical applications. We are going to have 5 different prestigious speakers, and let me introduce the first speaker, Dr. Dong-hyun Kim from Northwestern University. He will be presenting on translation and nanomedicine for interventional oncology cancer therapies. Everybody, please welcome Dr. Kim.
Okay, thank you very much for that kind introduction. So yeah, as you introduced, my name is Dong-hyun Kim. I'm an associate professor of Department of Radiology and Biomedical Engineering at Northwestern University. So I'm very excited, and I feel that I'm very honored to give a talk in the Big C 2023. Thank you for inviting me, and thanks for joining in my talk. So today I'm going to talk about the translation of nanomedicine for interventional oncology cancer therapy. Actually, that is what we are working in our lab. So today's presentation is including those contents. So I'll briefly introduce the current challenges in cancer medicine, and then I share that the promise of nanomedicine with the future direction, and also that I'll introduce our lab very briefly. Then I will talk more details on our specific project about imaging-guided cancer nanomedicine using multifunctional nanocarriers. I think everybody knows that the cancer is the second most common cause of death in the US, and the cancer cases keep rising in every year. So in 2022, 1.9 million new cases, and then about 610,000 cancer-related deaths were reported. And then worst situation is that by 2030, the number of the cancer cases will increase 45% more, and then about 800,000 cancer-related deaths can be happened. So it is indicating that the conventional those therapies, such as like a surgery, chemotherapy, radiation therapy, could not work well for treating cancers. So one of the main limitations on the conventional cancer therapies will be the low targeting efficiency. So surgery is commonly applying there are being margin during the surgical resection of the tumor, and the normal tissue damages happen a lot and they're almost unavoidable. And then chemotherapy is usually systemically applied, and those toxic agents are affecting to the all over the body. And the radiation therapy is using ionizing energy. So when the radiation is penetrating the body, all irradiated cells and the tissues will be damaged too. So those non-specific delivery of the therapeutics are generally causing many side effects like this all over the body, and then that resulting in stopping the treatment, any kind of treatment, and then overall survival from the cancer will be very low in many cancer cases. So innovative cancer therapy that can kill cancer cell and the tumors very specifically, that is strongly needed for minimizing cancer-related deaths. So one of the important approaches for that would be the cancer nanomedicine. So as you can see, the size scale of the nanoparticle is very similar with cellular component, and those size similarity and their high surface area of nanoparticle provides a good opportunity for the biomedical applications. Especially that the advancing of the nanotechnology that has found that the various new functions that can be used for medical therapeutics and the medical imaging. So also that they established the molecular linking chemistry that allowed the engineering of multifunctional nanoparticles. So as you can see here, that the nanoparticle itself that can work for therapeutics and the carriers and the imaging agent, and if necessary, that the various functional molecules can be attached to the surface of nanoparticle. So these nanoparticles have a great potential for the development of the innovative cancer therapy. So up to now, that the actual nanomedicine has demonstrated a huge potential benefits in the cancer diagnosis and then imaging and then therapy. So nanoparticles can refine and improve all essential procedure for the cancer treatment and then prove their efficacy. So as you can see here, that actually the nano product has been FDA approved and used in the clinic. And then here are the most updated information of nanomedicine. So about 100 nano products are currently available in the market for the uses in the clinic. And the promisingly that about 435 clinical trials are ongoing. So here are a little bit more detail about the developmental stage of nanomedicine. There are many like phase 1 and the phase 2 stage clinical trials. So it is indicating that the more nanomedicine, the products are coming soon. And then targeting indication of nanomedicine is mostly cancer. It is more than half of the indication is related with the cancer. Then, as expected up to now, that the repeat-based nanoparticle are the primary formulation for the translation of nanomedicine. That is quite understandable because the first FDA-approved nanoproduct was liposome carrier with doxorubicin, that the much clinical data that makes it much easier to get approval by the FDA, especially for many applications of the liposome-based nanocarriers. And then relatively new that the inorganic nanocarriers, actually that is more likely can achieve various multifunctional nanomedicine. It is still early stage, but about over 16 inorganic cancer nanomedicine products are available in the market. And then those are including iron and the gold and then hafnium nanoparticle. And then more promisingly that more than 70 inorganic-based nanomedicine are also in the clinical trials too. So it is a very promising trend with significantly increased the clinical trials, and also that we recently witnessed the successful story for the COVID-19 vaccine using nanocarriers. So now we can expect that many more highly efficient nanocarriers, nanomedicine can be available in the clinic near future. So more promising things with those potential of the nanomedicine, the federal funding and the support from the NIH has been significantly increased in these days, and especially in the cancer research. And the new special initiatives and the programs for promoting that the clinical translation of nanomedicine has been established very recently. And at the same time that the many startup companies and the developing nanomedicine could raise the significant funding from the venture capital and the investor and the industry partnership. So with that, the FDA are accepting significantly increased the amount of application from the company for developing the nanomedicine product. And at the same time that recently FDA could establish the various nanomedicine-related regulatory guidance. The most recently issued FDA guidance is titled with nanomaterial containing drug and the biological product for industry. It has been issued on April 2022. So I think it's truly now it is the translational stage of the nanomedicine to the clinic. And then here is another information about the promise of nanomedicine. So National Nanotechnology Initiative, the 2023 budget has been approved like this. So as you can see from the past, from 2001 to up to 2020, the cumulative that the U.S. government nanotechnology investment is 29 billion. And then the fiscal year 2020 year budget was 1.4 billion. But now the cumulative number of the budget is 40.7 billion. And then in this year, the budget is 1.99 billion. It is much more increase. So especially that as you can see that in the NIH budget, I think that is a significantly increased compared to the NSF, that is funding more fundamental science. So it can indicate that nanotechnology is going into more medical application and the clinical translational stage. And also that in the NIH funding trend, they are actually that the NIH covering the many different medicine area, but as you can see here, we couldn't notice that the cancer and the nanotechnology and the biomedical imaging could be the primary area in the NIH investment. And then one announcement from NIH is shown in here. So we can see that the NIH's effort on developing translational the nanomedicine for cancer intervention too. Then this year, very recently, that the NIH NCI that announced that the most promising area for nanomedicine applications. So these are the list, and the delivering chemotherapy is still one of the main area that nanomedicine can contribute, and also the next is delivering or augmenting the radiotherapy, and the nano-enabled immunotherapy and delivery gene therapy. Those are the most promising area for the nanomedicine in these days. Then, as mentioned before, that the many startups has been launched, and then actively working for commercial nanomedicine products. So here that about 305 nanomedicine startups all over the world, and then here that the 5 top leading companies are showing in here. So based on the current advance of nanomedicine, and we could see that this nanomedicine can be one of the most promising future clinical cancer medicine. Yeah, however, many people know that there are still many challenges on the translation of the nanomedicine to the clinic. So it depends on the developmental stage of nanomedicine. Those challenges can be separated in each developmental stages. In technology stage, in the large-scale synthesis of nanomaterials and the safety consideration, and then more importantly, that the development of purpose-driven engineering technique are very challenging. And also that in the biological part, actually that most of the nanomedicine is based on the systemic administration. But during the body circulation, various biological barriers are challenged to improve the targeting efficiency of nanomedicine for expected function of the nanomedicine. And also that in the clinic and the company, especially for focusing on the clinical trial and the commercialization stage, we think that the patient recruiting and the patient selection could be very critical. And also that there is a lack of the detail of the regulatory and the guidance that is very challenging to make move for the nanomedicine to the clinical translation. So in summary, that the current nanomedicine should be focused more on the translational stage, and that they can initiate the more clinical trial. And then to do this, and we should make more effort for finding very specific indication, and then disease-specific and the clinical needs-based engineering of nanomedicine is extremely required. And that's at the same time, they are developing clinically approvable targeting and the localization approaches that should be developed for the clinical nanomedicine. And those efforts will be connected to the various clinical trials and eventually that achieve successful translation of nanomedicine to the clinic. So our Big Med Lab is working for the potential translation of nanomedicine. So for the clinical needs and the clinical applicable targeting approaches, we are studying the clinical image-guided medicine. The story of the image-guided medicine is relatively short, but it is the most promising method for the future personalized medicine. The barriers that the preoperative and the intraoperative and postoperative imaging that has been used for the precise the cancer therapy delivery and the therapeutic response monitoring. So that could be the best clinical practice platform that can be comes with the medicine. So what we are investigating the clinical needs on the image-guided medicine, and then based on the specific clinical practices, we are designing and engineering nanomaterial, and then we are testing those in vivo, in vitro, those material for clinical feasibility and the efficacy of the image-guided nanomedicine. So here are our ongoing project for treating those solid tumors. So we are focusing on the various local delivered therapeutics and the ablation therapies as listed in here. So to enhance those therapeutic efficacy of each image-guided medicine, we design nanoparticles as shown in here and suggest new image-guided nanomedicine approaches. So we are actually testing that if our proposed image-guided nanomedicine can significantly enhance the image guidance capability and then therapeutic efficacy. So as you can see here, that our effort and then approaches is demonstrates a very promising result, and our result has been highlighted in many high-impact papers, and also that our importance, our research was approved with the multiple funding from NIH and the cancer-related societies. So here I'd like to introduce some of the project that we are working on, the image-guided nanomedicine. So as you may know that the immunotherapy is one of the hot topics in the cancer treatment. So in the previous slide that the NIH suggests that the nano-enabled immunotherapy is one of the most promising nanomedicine area. So we have focused on the image-guided local nano-enabled immunotherapy. So most recently, that published the paper. This paper is related with the enhanced local delivery of anti-PD-L1 immune checkpoint inhibitor immunotherapy. So in here, that we developed immune checkpoint inhibitor binding adapters equipped ion cell nanoparticle for local nanoimmunotherapy applications. And we also developed multifunctional nanocarriers for an efficient radio immunotherapy application. So developed a radiation responsive degradable gold nanoparticle that could deliver local anti-PD-L1 immunotherapy with highly efficient radiation therapy with this nanoparticle. And then another project I'm involved in the clinical transcarceral hepatic arterial chemoembolization. So we have developed some external magnetic field responsive the drug-eluting microspheres. So that can deliver the combination anti-angiogenic and then cytotoxic combination chemo agent in the local hepatocellular carcinoma with the intraarterial delivery. So it demonstrated that an enhanced therapeutic efficacy of the taste for the hepatocellular carcinoma. So in the following, the next two slides, so I briefly introduce the image-guided local nanoimmunotherapy project. The project title is the G-domain protein nanobiointerface MRI visible anti-PD-L1 nanoconjugate for enhanced local immune checkpoint inhibitor immunotherapy. Actually, it was published very recently, 2022 in Nano Today. Okay, this project is local nanoimmunotherapy, and then we focused on the hepatocellular carcinoma. So that is called the HCC. So HCC is the third highest cause of cancer-related deaths worldwide. So immune checkpoint inhibitor immunotherapy could be one of the very promising approaches. However, in the clinical outcomes showed about only 20 to 30% overall response rate. And that adverse side effect is pretty high. It's over 50%. So systemic administration, systemic administration of the anti-PD-L1 antibody and the following the low targeting efficiency could be one of the big reasons for the low therapeutic response rate. So actually the anti-PD-L1 antibodies, that is the IgG antibodies, the structural antibodies is mainly composed by the antigen-specific, the FAB regions and the FC region in tail. So when these IgG antibodies are injected systemically during the circulation, extensive off-target binding and then proteolytic clearances can happen. So that eventually reduce the the targeted PD-L1 inhibition in the tumor, and then most of them are actually that excluded so that the macrophage-mediated RDS clearance and observed in the spleen and the liver like this. And then even if that rest of them can be delivered with the EPR effect in the tumor, the actual delivered dose can be very low. So here we use the G domain, that it has been known that the IgG FG domain specific binding protein from the Staphylococcus aureus. So we engineered this G domain to control the orientation of this antibody on the surface of the nanoparticle. So we firstly engineered this G domain to be the GST attached to the G domain. So that GST G domain can be attached to the glutathione, the modified pheromoxytol, and then the G domain equipped nanoparticle can be easily attached to the anti-PD-L1. Then this simple the mixing of the anti-PD-L1 and the G domain, the equipped pheromoxytol, then makes this kind of oriented antibody attached to the nano complexes. So this anti-PD-L1 FAB region is exposed outside, and the FC domain hiding inside of this nanoparticle. So we hypothesized it that it will enhance the avidity of the anti-PD-L1, and then that can result in immune checkpoint inhibition. So for in vivo application, then we tested this with hepatic intra-arterial infusion for local immunotherapy of HCC in NRS1 HCC rat model. So local anti-PD-L1 infusion can be performed like this X-ray, the angiography imaging system, and an MRI. And then at 24-hour post-infusion of our sample, we can observe that the enhanced the local binding and the accumulation of the anti-PD-L1 nanoparticles. That is a compound with the highest Prussian blue staining in the tumor tissues. And if we see that the cytotoxic lymphocyte to regulatory T cell ratio, so IA infused that our nanoparticle shows that the highest value. So it is indicating that the successful activation of anti-cancer immunity. Then we measured the tumor responses after the IA delivery of our nanoconjugate. And then it was compared with those control groups, including systemic anti-PD-L1 injection, and then just IA delivered anti-PD-L1 immunotherapy. So as you can see that our IA-infused anti-PD-L1 G-domain pheromoxytol showed that the highest tumor regression in 15 days. So those developed nanomedicine can be further developed with those interventional oncology techniques. Actually, that is established for reaching many organ tumors. So as you can see here, that the liver and the pancreas and the heart and the prostate and the kidney and lungs, they can be targeted with this image-guided catheter positioning. And so well-designed nanomedicine for each purpose of this image-guided nanomedicine, that is, if that is combined, that could have a very high impact for cancer medicine. Okay, so here is the conclusion. So I think the image-guided cancer nanomedicine will be the key approaches that can significantly enhance the cancer therapeutic outcome, especially that it can be very beneficial for the application, specifically delivering chemotherapy and the radiation therapy and then immunotherapy and the gene therapy. So by making the effort much more on the clinical translation of the nanomedicine, I hope that the image-guided cancer nanomedicine can be used as a strong tool to overcome the current limitation of a conventional cancer therapy. Okay, thank you very much for all our lab members, and especially I really appreciate that all the collaborators and especially MD medical doctors, they provided the various clinical views that the nanomedicine, and also they just provided the very specific some the prospect for the clinical practices of the nanomedicine. And also that I really appreciate all the funding agencies, including the NIH and NCI. Okay, thank you very much.
Thank you for your valuable presentation. Now we're going to have some questions from the audience. Okay, Dr. Kim, I have a quick question. So there are many companies and researchers are still struggling to get the final FDA approval for their nanomedicine, especially in the field of the cancer therapeutics, and only limited amount of the products will be available in the market worldwide. So can you please give us your opinion and insight about the most important criteria for the successful FDA approval, mass manufacturing, and the final clinical applications in terms of the biomaterial design based on your research experiences?
Okay, yeah. I think that my presentation is all part of those kind of like answers. So actually there are like many like potential nano product actually that has been studied and then published in the many papers. But the commercialization and then like clinical translation can be very challenging because I think first of all like safety issues that can be very important. So those safety issues should have like a big amount of like clinical data, and then also that the in depth of that the toxicology test. I think that should be done before the clinical translation. So for doing that, I think that's why that we are more focusing on the conventionally already used in the material, like already approved, FDA approved the material, like ion oxide nanoparticle, that has been used in the human body, and also that there are other like polymeric material that is already proved that their uses in the human body. So utilizing those materials can be much better for the rapid process for that like some kind of FDA approval. But I think even if the new materials, maybe if we can thoroughly test the like toxicology with like FDA or like the NCI that they have like their own lab for evaluating those like the basic toxicology test. So maybe that we can apply also to do that. And also like after that, we need to think about the GLP, the facility, and then how to make that like a high yield process. Those should be followed for the commercialization. So there are so many considerations. But I think based on the commercial product, maybe a little bit safer and the quicker way is better for like expecting more than the nano product.
All right, thank you so much.
Mm-hmm.
And now we're going to move on to our next speaker, Dr. Seung-Pyo Hong from University of Wisconsin Medicine, and he will be discussing about the dendritic nanoparticle for targeted cancer immunotherapy and liquid biopsy as its companion diagnostics.
All right, well, it's my great pleasure to be here. I thank the organizers and session chairs for really kind introduction as well as a kind invitation for me to be allowed to come back to my own country and then enjoy the people and culture, everything. So is it showing up? So today I will talk about a couple of our projects that we are working on in our lab, mostly focusing on like Dr. Kim mentioned, nano-enabled immunotherapy as well as nanotechnology-based liquid biopsy. So because of, thanks to Dr. Kim, who really gave a great talk about the backgrounds and then recent progress in cancer research and NIH support, I feel fortunate that I'm gonna spend more time focusing on my own research. So full disclosure, so I'm working with multiple private sectors and companies. There's a Capio Biosciences that is trying to translate our liquid biopsy technology ultimately to the market, which is now based in the USA as well as in Korea, here in Songdo, by the way. I'm also working with the Dongguk Pharmaceutical in Korea, trying to translate one of our projects, and then I've been serving as a consultant for a few years now. I'm also working at BioView in Israel, as well as Warfone in Spain as a consultant. So our ultimate goal as cartooned here is actually a big vision that I've had over the years. So ultimately, we really want to achieve some truly personalized medicine by developing a nanoparticles that can deliver multiple drugs in a controlled and in a targeted way, as well as some liquid biopsy technology where we can quickly give the feedback to the formulation teams and physicians so that they can actually choose the best performing medicine combinations. So briefly speaking, so we are working on multiple platform technologies as shown here that can deliver, so right now the current trend to the cancer therapy, especially mediated by nanoparticles, is all about combination therapy, because a number of clinical reports show that the single modality-based treatment is usually not effective enough. So we are trying to develop those combination therapy platforms and apply to individual patients. And then the way that we monitor how that particular patient responds to that particular combination is going to be through simple blood draw, where we are trying to develop blood circulating biomarkers, such as circulating tumor cells or tumor-derived exosomes, to be used as a reliable biomarker, and then that can provide the information back to this team. And then if that works, we use the same combination. If it doesn't, we use we change the combination. So eventually, we want to make this type of closed loop system, if you will, to really realize truly personalized medicine. We are not there yet, though, but we are trying to get there. So I've been showing the same slides over and over, so some of you guys may have seen this, but over the years, we have been interested in some of the nanomaterials, particularly that have some of the dendritic structures. So we started working on dendrimers in the beginning a couple decades ago because of some of the advantages for the dendrimers to be applied for a variety of biomedical applications, because as listed here. But one of the major reasons why we are particularly interested in dendrimers and dendritic structure is because of their ability to mediate what is known as multivalent binding effect. What it does is that, this is actually happening in nature all the time. So we all just went through the COVID-19 situation. So when a virus attaches to the host cell, they always induce multiple binding pairs simultaneously. So the simple argument is that if you induce 5 simultaneous binding pairs in a highly localized area, the energy required to detach all 5 at the same time is going to take orders of magnitude stronger energy than the energy required to detach 1-to-1 monovalent binding counterpart. And then by doing that, you can really manipulate, engineer the binding kinetics of various ligands mediated by the dendrimers or polymers. In experimentally, we actually have shown that we can decrease the KD value by up to 1,000,000-fold, meaning that a 1,000,000-fold enhancement in terms of binding strength. And this is a really old computer simulation where it shows that the dendrimer is flexible and deformable enough so that it's one of the ideal mediators for multivalent binding effects. So that's what we've been working on. So more recently, we've been particularly interested in cancer immunotherapy, as all probably of you know, that this is really changing the paradigm of the cancer treatments right now. And then among the many immune checkpoint inhibitors, we have been really focusing on PD-L1, PD-1 pair, and then trying to develop some new technology that can more effectively intervene that PD-L1 and PD-1 binding. So briefly speaking, if the tumor cells overexpress the PD-L1, they can bind to PD-1 that is often expressed by the T cells, and that binding actually results in inactivation of T cells so that the T cells don't recognize the tumor cells as the subject of attack anymore. So this is a well-known mechanism of cancer to evade the immune attack. But if you disrupt this binding by treating the cells with either anti-PD-L1 or anti-PD-1, you can block this binding. And if this binding does not occur, now the T cells can be activated and then they can kill the tumor cells. So people have been working on this PD-L1, PD-1 immune checkpoint blockades for many years. And then, as you know, a few years ago, the Nobel Prize was given to those folks who developed this. So the first thing we did was that we have been working on the dendrimers and working about, kind of enhancing the binding kinetics. And then the natural evolution of our idea was about using the antibodies that are available in the clinic already. And then through the combination of those antibodies, the nanoparticles, we can significantly enhance the immunotherapeutic efficacy by doing that. And then at that time, I had a fantastic postdoc, Dr. Ji-Yoon Boo, who stayed with us for about 4 years, and then he recently started his own lab at Inha University here in Korea. So our idea was that we just conjugate the multiple anti-PD-L1 antibodies to the dendrimer, and then that will result in enhancement in binding kinetics, and then that can translate into enhanced T cell activities and eventually decrease the tumor cell survival. And then I'm gonna skip all the data that Ji-Yoon produced, but we actually did a lot of studies and measured the binding kinetics in vitro and in vivo, and we got the very promising results. And then later we became more interested in kind of replacing those antibodies with something more simple or easier to manipulate or easier to synthesize. So a vast majority of the clinically available immune checkpoint inhibitors are antibodies, if not all. They have distinct advantages, such as they are proven to be effective, efficacious, and then they're proven to be clinically effective, which is very important. The problems come in because antibodies are really big, so we're talking about 150 kilodalton molecule, and then they are very expensive. So if you're running a lab, buying 100 microgram of antibody can easily cost up to $500, if not more. And then they are hard to engineer, and then also some of the reports indicate that there are large batch-to-batch variations. So even really interested in the peptides as an alternative. So peptides, on the other hand, have many advantages, such as you can really easily modify and synthesize the molecules. They are smaller, so me as an engineer is a lot easier to play with. They are significantly cheaper than antibodies. However, the problem of peptides, and that's the major reason why peptides have not been really making into the clinic as nearly as much as the antibody, is because of their weaker binding, significantly weaker binding compared to the antibody counterparts, and their low retention and short half-life in the body. So we are trying to address that through the combination of the peptides and nanoparticles. And then at that time, another fantastic postdoc joined my lab, Dr. Woo-Jin Jung, who also ended up becoming an assistant professor at Inha University. So there's no connection, by the way. But both of the postdocs ended up working for Inha. I think it's actually lucky for Inha. And then, so he came in, and then he was a peptide expert. And then he and I talked about how to really synthesize and manipulate the peptides to be appropriately present on the surface of the dendrimers. And then we designed this by looking at the PD-1 antibody and then taking out the binding sequence out of it so that they can target the PD-L1. First thing we did was we measured the binding kinetics. So long story short, so if you compare the free peptides, you're talking about the KD value of 10 to the negative fourth molar, which is fairly high, meaning weaker. And then if you compare that with our conjugates, with a dendrimer with the multiple copies of peptides, PD-L1 binding peptide, we enhance the binding kinetics by 5 orders of magnitude, which is significant, which is at the same level with antibodies. And nowadays, we are actually getting even better results of dendrimer peptide conjugates binding even more strongly than the antibodies as well. And then we recently studied the in vivo study to finally, assess the in vivo efficacy of the materials that we engineered. And one of my graduate students, Piper, has been leading the synthesis of these peptide sequences, among other immune checkpoint inhibitors. But I'm not going to talk about others today because of the time constraints, and then focus on the PD-L1 binding peptides. And then this is one of the recent results that we got from in vivo study using syngeneic mouse model that has the MAC1 tumor cells in the immune competent mice. So Daewon is another Korean student who is working in our lab right now, did this all in vivo studies and found out that upon injection, we observed immediate reduction in tumor size. Remember that this is actually single modality-based treatment. So we got very excited of seeing this much of efficacy using just one immune checkpoint inhibitor, and then there's no sign of toxicity. We also did a lot of biological testing afterwards, and including histology and H&E staining. But today I just wanted to show our nanostring data that really analyzed the tumor tissue after the treatment with the dendrimer peptide conjugate. We call them DPCs, in terms of their upregulation of certain genes as well as downregulation of certain genes. So as you can see here, we identified a number of genes that are upregulated as a result of either DPC treatment or antibody treatments. We got fairly similar results between the antibodies and our conjugates. And then you see that the upregulated genes are highly associated with T-cell activation, adaptive responses, innate immune responses, inhibitory immune checkpoints, and cytokines. On the other hand, we also identified a number of genes that are downregulated as a result of the treatment with the antibodies or DPCs. And then those downregulated genes were closely associated with the cytokines, adhesion, inflammation, as well as cancer progression, showing that we are kind of like upregulating genes that we want them to be upregulated, and then downregulate the genes that we want them to be downregulated, so that we can really maximize the efficacy of our treatment options. In addition to those dendrimer-based peptide conjugates, we are also making some dendromycels. This is going to be for combination therapy, because not only we can carry the peptides on the surface of these nanoparticles, but also we can encapsulate the chemo drugs in the core, so that we can combine those together so that we can maximize the efficacy. And then because of time constraints, I'm not going to talk about that much today. And then not only the cancer, we are also interested in pulmonary fibrosis, trying to use our dendromycels as delivery vehicles that typically treat the fibrosis fairly effectively. And then lastly, we've been also developing some lipid nanoparticles. Everybody is talking about lipid nanoparticles nowadays. But the uniqueness of our approach is that we use the dendritic components into the lipid nanoparticles, and then we have shown that we can enhance the gene delivery efficacy without apparent toxicity. So we are actually moving forward with this, and then all of these are going to be used for combination therapies, as I mentioned in the beginning. Switching gears, I wish I had more time, but I only have 10 minutes left. The other side of the lab has been working on the liquid biopsy technology development, as I mentioned in the very beginning. So the two target markers that we are trying to develop are circulating tumor cells as well as tumor-derived exosomes. And then again, the COI full disclosure with Capio that I co-founded, and then I'm serving as a CEO and president. So liquid biopsy has emerged about 2 decades ago, because the current way of diagnosing and monitoring the cancer patients right now is either through the solid biopsy, which involves the optical operation and very invasive, and then the other is going to be the imaging based on PET CT scans and MRI. So obviously the tissue biopsy can cause a lot of problems, and the invasive nature can even cause cancer metastasis as well. And then not every tumor is accessible by surgical operation. So this is actually limited in that way. On the other hand, PET CT scans and MRI are fantastic imaging technologies, but they are expensive, and probably some of you may have been imaged. If you're getting the MRI imaging, for example, you have to go into the really small tube that barely fits your body, and then stay there still for 40 minutes with incredibly loud noise that I hate. So that was not a really pleasant experience. So instead of doing that or supplement of those technology, one can consider taking the just blood out of the patients and then go through some device and then measure their biomarkers circulating in the blood. And then if they can provide the same or the better information, that's going to be beneficial for not only clinicians and patients, obviously, and mostly patients. So they are cheaper, faster, and easier. There are several biomarkers that people have been really focusing on, including cfDNA, cell-free DNA, cancer exosomes, as well as CTCs. So among the three, briefly speaking, the cfDNA has been implemented in the clinic fairly nicely right now. It has most advanced stage in terms of clinical utility. However, recent reports actually indicate that just cfDNA is just not enough because there's some issues that I'm not going to go through so much of details, but they are like cell-free DNA, and then nobody knows when the DNA was actually released from the cells, and then it doesn't really reflect the dynamic status of the patient. And also the information we can pull out from the cfDNA is actually fairly limited compared to especially the CTCs, which is on the other end of the spectrum. So capturing the whole cells, we can pull out a lot more information, DNA, RNA, and proteins, and full genomic sequencing as well. The problem is it's a lot harder to capture and analyze compared to cfDNA because of technical difficulty. So some of my lab members, Dr. Ja Hae-myung, who used to be our lab for really long. She was my very first PhD student and then did a postdoc with me as well, now working for FDA. Mike Pullman, he's my lab manager right now. He's been doing the postdoc and the long-time research scientist in the lab. And Ji-yoon at Inha also contributed to this project quite a bit. So to address those technical challenges, we developed a capture device that has really high sensitivity and specificity, along with the expandability to accommodate multiple cancer cell markers so that we are not limited by just single markers that we can rely on. Basic idea was we wanted to mimic what is happening in the blood vessel wall in our device. So, briefly speaking, we wanted to induce the cell rolling on the surface of our capture surface by immobilizing the selectins. So what it does is that it can effectively recruit fast-flowing cells to the capture surface by not inducing the abrupt adhesion, but inducing dynamic rolling response of the cells. And cells are flowing fast, and then meet with those proteins, and then started to be recruited on the surface and roll along the surface. And between those rolling cells, we can capture the CTCs more specifically using those dendrimer-mediated multivalent binding effect, as I mentioned, through the immobilization of the dendrimers with the multiple kinds of antibodies and peptides, of course. So we've published a few papers, as listed here, to validate the device designs and engineering, as well as in vitro and in vivo studies. And most importantly, we've done quite a bit of clinical studies over the past few years. So I'm going to introduce just a few of those data today. So this was the clinical study that we did in collaboration with the University of North Carolina, Chapel Hill, where we recruited the head and neck cancer patients. And then in this particular study, we divided those patients into two groups. So groups on the left shows that they are responding to radiotherapy in this case really nicely. And then the group on the right-hand side is the patients who show the progressing disease, meaning that their prognosis is not really good. And at the same time, if you really compare the CTC kinetics, we saw that the CTC number went down dramatically and stayed low for the patients who were responding, versus the CTC numbers were continuing to go up for the patients who are not responding and having the progressing disease. And then if we look at those patients individually, this is one of the patients with the head and neck cancer. So this is a PET CT scan where we are looking at this particular area. So if you look at the bright spot here with a sharp contrast, meaning that there is a tumor indication, and this patient with the tumor indication right there, with a relatively high number of CTC, with the treatments, they went down, and then after the treatments, about 1 month after the treatments, it started going back up. And then we imaged the same patients with a PET CT scan, and then we saw that most of the tumor was gone. So there was some discrepancy right there. And then as we monitored this patient longer, 4 months after the treatments, the CTC number continued to go up, and then in the imaging, the tumor obviously came back. So showing that even the CTC as a biomarker has potential to be used as a predictive biomarker as well, compared to the imaging based on this data. These are other patients with a clear indication of the tumor with a high number of CTCs. And these patients actually responded to radiotherapy really nicely, so that the CTC number went down and continued to go down. And then 9 months after the treatments, the patients got the complete response decision by the clinicians. And this is the most recent results that are about to be published. maybe today or tomorrow, I don't know, at any time right now. So this particular study, we did a study with in collaboration with the UT Southwestern as well as the UNC Chapel Hill, where we recruited oligometastatic cancer patients, meaning that those patients have a few metastatic lesions. And then we divided those patients in two groups based on the CTC trend, right? So the blue lines indicate the patients who showed this overall decrease in CTC numbers over the treatments, and then the red lines indicate that the patients who showed the overall increase in CTC numbers over the treatments. And then we divided them into the two groups and compared the survival of those patients, and we got really statistically significant results where those red line patients actually showed much shorter survival compared to the patients who showed the general trends of decreasing CTCs. So we got excited and we published this, about to publish this in JCO Precision Oncology. Not only we can capture and counter cancer cells, but we can also stain them with various markers, P63, PD-L1, and CMET as an example. And then we recently did also the single cell RNA full sequencing based on the CTCs we captured, and then identified about 68 cells from a single patient as a CTC-like cells based on the full sequencing of our single cell RNA. So I hope that I presented like somewhat convincing data that we are trying to stem from the dendrimers and making some various nanoparticles for effective drug delivery, as well as using the dendrimers and biomimetic technology to detect the biomarkers, especially those circulating in the blood, and then try to develop those CTCs. And I didn't have time to talk about the exosomes, but we are also working on the exosomes as a secondary biomarker as well. So I think this is the most important slide that I present today. So I've been really fortunate to work with fantastic young individuals who are highly talented, like both graduate students and postdocs and staff scientists. And then I've been really fortunate to work with a fantastic lineup of collaborators as listed here. And then I've been extremely thankful of those funding agencies that have been supporting our research financially over the years. So with that, thank you for your attention. I'd be happy to answer any questions. We only have 33 seconds though.
Thank you for your presentation. Any questions from the audience?
Okay, I have a simple question.
Sure.
Thank you for the great talk, Professor
Thank you.
Hong. I enjoyed a lot. So I'm wondering how much improvement you've achieved with your peptide epitope-based approach in terms of engaging PD-1,
Sure.
as well as anti-tumor therapeutic efficacy compared with the antibody.
Sure.
approach.
Well, I didn't actually include that data. So we recently did a comprehensive in vivo studies comparing the free antibodies versus our conjugates. And then we saw that, so this is actually fairly well known. If you repeatedly inject anti-PD-L1, especially through IV, there are some well-known toxicity involved. The mice actually die after so many injections. We didn't see that toxicity from the DPCs, which is a really good sign to begin with. And more importantly, we got better efficacy about, I can't remember the exact number off the top of my head right now, but fairly significant enhancement in terms of efficacy improvements using our DPCs. And those effect was further pronounced when we actually employed multiple immune checkpoint inhibitors. So we are also working on not only PD-L1, but PD-1 and TIGIT, among others. And then whenever we put multiple targets, we typically get much better in vivo efficacy results. Thank you.
If there is no more question, we are closing the second speaker's talk. Thank you very much.
Thank you.
Our third speaker is Dr. Yong-woo Cho from Hanyang University. He is also a CEO of Exostem Tech, one of the most successful company to develop the exosome therapeutics product. And he will be presenting on taking exosome therapeutics from university lab to clinical trials.
Thank you. Thank you for kind introduction, Chairman. I'm Yong-woo Cho, the professor of Hanyang University, Erika campus. and also the CEO of ExoStemTech. 7 years ago, I found a small bioventure company. The company name is ExoStemTech, which means exosome stem cell technology. The meaning is pretty straightforward. Currently, we are developing several kinds of exosome-based biopharmaceutical for treatment of osteoarthritis, lung fibrosis, and tendinitis. Today, I'm going to share my research experience on taking exosome therapeutics from academia to industry, from university laboratory to clinical trial. Here is the exosome. Almost all cells secrete extracellular vesicle. There are known to be 3 kinds of extracellular vesicle. The largest one is apoptotic body. The middle one is microvesicle. The small one is exosome. Sometimes we call it small extracellular vesicle. Exosome carry nucleic acid and protein and diverse types of metabolite. Here you can see here CD81, CD9, and CD63 are well-known exosome surface marker. Well-known exosome surface marker. The main component of exosome, phospholipid and protein and RNA. This review paper explain the main role of exosome, such as regulation. Exosome are involved with the regulation of gene transcription and translation, survival and proliferation, reproduction and development, and so forth. So exosome are kind of messenger for cell-to-cell communication. Here you can see the well-known coronavirus. The coronavirus, the virus is the most effective gene delivery system evolved by DNA delivery system and RNA delivery system evolved by nature over the eons. You can see some similarity between virus and exosome. Their sizes are similar. The exosome size are in the range of 50 to 150 nanometer, and the main components are similar, phospholipid, protein, and RNA. So considering that, if we load therapeutic protein or therapeutic biomolecule inside the exosome can be very effective drug delivery system. Here are our main exosome pipelines. We are currently developing several kinds of exosome, stem cell exosome. Here is the cartisome for treatment of osteoarthritis. Here is the tendinosome for treatment of tendinitis. Here is hypoxisome for treatment of lung fibrosis. Also, we are developing some platform technology which can load therapeutic protein inside the exosome. The red one is exosomal surface marker, for example, CD9. And the green one is photocleavable linker. And the blue one, a violet one, is therapeutic cargo. So by using genetic engineering technology, we can load a diverse type of therapeutic protein inside the exosome. This kind of exosome for treatment of tendinitis, Parkinson's disease, or Alzheimer's disease. Also, we are developing RNA delivery system as a carrier, the use of exosome as a carrier of microRNA for treatment of diverse inflammatory disease. Also, we are currently developing a plant exosome. Plant actually secrete a large amount of exosome for the use of cosmetic or dietary supplement. Also, as you know, the cancer cell exosome play a significant role on cancer cell proliferation and cancer cell metastasis. So we are developing cancer cell exosome, cancer exosome inhibitor to inhibit the secretion of exosome from cancer cell. Okay, here is the main slide what I want to talk about in this presentation, taking exosome from university laboratory to clinical trial. First of all, the strong basic research data in terms of toxicity and efficacy are clinically important. 3 university laboratories collaborate to obtain promising exosome candidate to obtain a strong efficacy and safety data. The first university laboratory is my lab, Hanyang University, and second is Professor Dong-Gyu-jo Laboratory from 성균관대 University, School of Pharmacy, and Professor Jae-hyung Park Laboratory from 성균관대 University, School of Engineering. So we use to investigate toxicity and efficacy of exosome candidate. We use different in vitro model and different in vivo animal model, and we select some important, some good, promising exosome candidate, and then we have to design mass production system for exosome production. Some production steps which are not suitable for mass production should be replaced by suitable mass production step. And then we have to find out CMO company. Here is the very important keyword from translational medicine. And then we have to find out CMO company. You know, the small bioventure company and university, they do not have a GMP facility because GMP facility require a large amount of money. So we have to find contract manufacturing organization, CMO company, for exosome GMP production. In our case, we have 2 CMO company, Cha Biotech for cell bank construction, master cell bank and working cell bank, cell production, and then the second CMO company, our second CMO company is Korong Biotech for exosome mass production and for exosome mass production. And then we should transfer technical transfer all methods on exosome production and physical and chemical and biological characterization method. So all how to produce exosome, how to characterize, how to analyze exosome chemically, biologically, and physically, all methods should be well documented as a form of standard operating procedure. Also, APV means aseptic procedure validation. So all exosome production should secure the absence of mycoplasma, endotoxin, foreign virus, animal virus, etc. Also, quality control is very important issue. And in the toxicity study, some toxicity study should be performed in GLP, good laboratory practice. So single injection toxicity, multiple injection toxicity, immunogenicity, tumorogenicity, and safety pharmacology, cardiovascular system, respiratory system, and so forth. So also all these keywords are important for translational medicine. This is our first pipeline, cartisome stem cell exosome for treatment of osteoarthritis. Here is target indication, osteoarthritis, KL grade 2 to 3. The compositions are exosome derived from allogenic adipose-derived stem cell. The cell source is human fat tissue. Okay? The administration is 3 times multiple injection into articular cavity. 3 times intra-articular injections. The formulations are exosome solution containing 0.5 Trehalose in cell line. Trehalose is kind of a freezing stability enhancer. The exosome can be stored minus 70 degrees Celsius up to 12 months. Primary outcome is going to be pain relief and inflammation alleviation and cartilage protection and repair. Here is the clinical sample for clinical trial phase 1. This slide explains overall production and quality control, cartisome production. The first of all, we have to construct cell bank, larger amount of cell, okay? Master cell bank and working cell bank, as I told you, the CMO company is Chabiolab. Here is the quality control item for MCB, WCB. First of all, we donate we get human fat tissue from young and healthy women in the age of 25 to 35. The amount of fat tissue is around 150 millimeter. And then we isolate stem cell from human fat tissue and we construct master cell bank and working cell bank for exosome production. And here is the quality control item for MCB, WCB, appearance, sterility, cell quantity, cell viability, differentiation potential, and as I told you, endotoxin without mycoplasma, adventitious virus, karyotyping, STL, and in the absence of bovine virus and human virus. All these items are for the quality control. And then the second CMO is Korong Biotech. Okay? We isolate stem cell exosome from the conditioned media through 2 times of normal filtration, 1 times of tangential flow filtration combined with column chromatography. Here you can see the quality control item for exosome production, the appearance, pH, sterility, endotoxin, and particle concentration, and exosomal protein concentration. Also, the correlation between particle concentration, exosome concentration, and exosomal protein concentration can be used as a quality control item. Also, the correlation between protein and RNA, the amount of protein, exosomal protein, and the amount of exosomal RNA, the correlation can be used as a quality control item. And purity and potency and residuals, leaky test, operational volume test, insoluble particle test, all these are quality control items. Currently, we are producing from one batch more than 5 multiply 10 to the power of 12 particle in one lot. Expected dose in human clinical phase 1 trial is going to be 3 multiply 10 to the power of 9 particle per injection. So in one batch, in one lot, per one lot, we can produce more than 1,000 vial. Here you can see the clinical sample, GMP production, produced in GMP for cartisome clinical trial. Currently, we established 10-liter scale exosome production process. Here is the summary of how to characterize, how to control the quality of exosome. In terms of safety and physicochemical characteristic and identification purity and injection characteristic. Safety management involve aseptic test, endotoxin, animal-derived virus, donor-related virus, and confirmation of cell genetic stability, and so forth. And exosome characterization include exosome concentration measured by nanoparticle tracking analysis. The exosomal protein concentration are measured by ELISA assay. Also, the correlation between exosome concentration and protein concentration are critically important. Also, exosome shape can be observed by cryo transmission electron microscopy. Also, single particle dispersion can be observed by zeta surface potential. Usually, exosome are well dispersed singly. Zeta surface potential values are in the range of -20 to -30 millivolt. But some kind of aggregation, the zeta surface potential will increase. It's just like 10 millivolt like that. Also, as I told you, CD9 and CD63 and CD81 is exosomal surface marker, and GM130 and carlexin is non-exosomal surface marker. It's kind of Golgi marker or ER marker, things like that. We analyzed the exosomal surface marker through flow cytometry and Western blotting. Here, this slide shows the quality control steps. We performed the quality control 7 steps. The first one is MCB, WCB, intermediate 1, intermediate 2, and intermediate 3, drug substance, and drug product. All 7 steps, the quality control should be performed in terms of potency, purity, identification quantity, and sterility and safety, and excipient control. In my research, in my experience, how to validate the quality control item and quality control method is pretty hard work. And then we have to characterize exosome cargo in terms of protein and microRNA, RNA, and repeat. So exosome cargo analysis has been done by LC mass, NGS next generation sequencing, and cytokine array, microarray, ELISA assay, combined with bioinformatics. And then we have to connect the exosomal internal cargo to mode of action. So here is some example of a cargo mapping. Some kind of internal molecule are related to cartilage regeneration, and some of them are related to M1, M2 macrophage polarization. M1 is pro-inflammatory macrophage. M2 is anti-inflammatory macrophage. The exosome induce M1, M2 macrophage polarization, or MMP inhibition or Wnt signaling, things like that. Also, we have been using different in vitro model and in vivo animal model to validate the efficacy of exosome. Here is one example. Exosome control the inflammatory factor and inhibit cartilage degradation. So exosome contain different kind of biomolecule, such as TIMP1-2 and HGF, IGF, interleukin-4, and interleukin-10. Specifically, TIMP1-2 contribute the inhibition of MMP activity. So exosome downregulate inflammatory factor in human chondrocyte in vitro animal model. Human chondrocyte obtained from human osteoarthritis patient. Here, exosome treatment significantly downregulate MMP1, MMP3, MMP13, and also collagen type 2 alpha 1 significantly increase after exosome treatment. Also, exosome suppress pain factor in an in vitro human chondrocyte in vitro experiment. and TRPA1 and COX-2 interleukin-4 was significantly decreased after exosome treatment. Here is showed the exosome effect on protection of cartilage by exosome in MIA-induced osteoarthritis rat model. Here we can see here collagen type 2 positive area significantly increased after exosome treatment. This means, exosome. This means this result show the effect of exosome on cartilage protection by inhibiting degradation of cartilage matrix and regeneration of cartilage tissue through induction of cartilage matrix synthesis. Here is showed another in vivo animal result using MIA-induced OA rat model. Here you can see here OARA score. The OARA score was decreased in a dose-dependent manner after exosome treatment. And we have been using different animal models. Here is the in subacute arthritis model and in chronic arthritis model. Here you can see here in subacute arthritis model, exosome greatly improved cartilage tissue regeneration, okay? And very low marking scores after exosome treatment. And also, the exosome stimulate chondrocyte proliferation and movement. Here, the exosome promote the proliferation and migration of human chondrocyte obtained from human OA patient. Also, exosome inhibit inflammatory cytokine and induce polarization, M1, M2 macrophage polarization, like here. You can see here M1 macrophage and inflammatory cytokine decreased after exosome treatment, and M2 macrophage are increased after exosome treatment. This is another in vivo animal model. This model is destabilization of medial meniscus. It's kind of a surgical operation, OA model by surgical operation. Here you can see your OARSI grade significantly decreased after exosome treatment in DMN mouse model. This is the ACLT-induced OA rat model. Exosome show great efficacy, and MMX-induced OA rabbit model, exosome show a good efficacy for regeneration of cartilage. Here, this slide shows the summary of toxicity. Toxicity study, single injection toxicity, multiple injection toxicity, in vitro, in vivo tumorogenicity, and in vitro, in vivo immunogenicity. The summary of toxicity study is that no observed adverse effect level is just to be 2 multiplied 10 to the power of 9 exosome particle for lat joint. And next slide shows the summary of safety pharmacology in terms of central nervous system, cardiovascular system, and respiratory system. For central nervous system, we used the rat, and for cardiovascular system, we used dog and monkey, and Hager-Say implantable telemetry, and for respiratory system, we used mouse. The summary: no observation in behavioral change, no effect on heart potassium channel, no effect on respiratory rate and capacity up to high dose. Currently, we are studying a donor-to-donor variation. So we are we construct 3 different master cell bank and working cell bank from 3 different donor because donor-to-donor variation are great concern. So we studied repeat profile. We performed repeat profiling and cytokine profiling and protein profiling and microRNA profiling. Up to now, there are high similarity in exosome characteristic derived from different donor-derived adipose-derived stem cell. Here is the second pipeline, tendinosome, for treatment of tendinitis. And here is the hypoxisome for treatment of pulmonary fibrosis. They are all stem cell exosome cultured in different culture conditions. This is the platform technology. We can load diverse type of cargo protein, therapeutic protein inside the exosome using photocleavable linker. Here is the radionis exosomal marker, photocleavable protein, and therapeutic marker, therapeutic protein such as superrepressor ICAPA-B for treatment of inflammatory disease or PrOC for treatment of Parkinson's disease, things like that. Here is the photocleavable protein M-maple. The M-maple can be cleaved irradiation of light with a wavelength of 405 nanometer. So the free therapeutic protein can be delivered inside the recipient cell like this. Currently, we are developing exosome-based biopharmaceutical for treatment of neurodegenerative disease and inflammatory disease. This is the end of my presentation. Thank you. Thank you for your attention.
Thank you for the talk. And now we are going to have some questions from the floor. 네.
Thank you. Very good talk. I have a very quick question. So your first product item is related with arthritis. So the usage is that you use the multiple injection of the intra-articular injection of your product, right?
That's right.
The reason that what is the reason for the multiple injection? I think about the exosome is not remain in the areas, defect areas for a long time, or what's the reason for the multiple injection?
Frankly speaking, the medical doctor recommends 3 times multiple injection. The medical doctor, we need, he said, one-time injection is, anyway, the medical doctor recommends 3 times multiple injection for treatment of osteoarthritis. It's like, we have both single-time injection and in vivo animal model. We have efficacy, both single-time injection and multiple injection. Sometimes we inject 10 times. Yeah, we have positive effect. They showed positive effect, but medical doctor, they said to me, one-time injection, and then three months later, 2-time injection, and then 6 months later, 3-time injection, third injection, things like that. Okay.
I also have a quick question. So thank you for sharing your wonderful journey from obtaining the research outcome to the launching the commercially available exosome therapeutics. And what was the most difficult step for the entire transition process? For example, like finding out the CMO company or like launching initiation of the clinical trials, whatever?
Okay, the most difficult step, I guess, to get approval from a regulatory affair. Like, most of FDA, they do not know about, exosome or extracellular vesicle, how to, how to define exosome, and how to characterize exosome, how to control the quality, things like that. So, and then the second one is mass production. We have to find out the CMO company, but most CMO company, no, at that time they are not interested in exosome.
Okay,
Thank you.
thank you so much.
Okay. Hello, everyone. My name is Sejin Son from Incheon University. Moving on to next speaker, I'm really excited and I'm really honored to introduce next speaker, Professor Won-Jong Kim. He was my supervisor for PhD. So he's a professor in the Department of Chemistry at POSTECH, and he's talking about control of nitric oxide for the treatment of inflammatory disease. Please join me welcome Professor Won-Jong Kim.
Thank you for your very kind introduction. I'm very nervous because my ex-student introduced me. And anyway, today I'd like to talk about my recent research. As other speakers said, I have also my own startup company, and I want to talk about the academic work in my lab as well as the development process using the nitric oxide work. Mainly, I want to talk about the controlling of nitric oxide for treatment of inflammatory disease. So, as you know, nitric oxide has a very different role. At the very low concentration of nitric oxide, it can inflate the blood vessels, and it is very good for wound healing. However, at high concentration of nitric oxide, they can kill the bacteria and they kill the cells. So it has some anti-cancer or antibacterial activity at high concentrations. So we have to control the concentration of nitric oxide for the applications. So in our lab, sometimes we are developing nitric oxide releasing material, or sometimes we are developing nitric oxide removing materials. So sometimes we are developing nitric oxide releasing materials like a nitric oxide donor. But sometimes we have to remove nitric oxide from the inflammation site. In that case, we are developing nitric oxide scavenging materials using the small molecules. But all these small molecules is not available by itself because it has very low biocompatibilities. So we have to conjugate or we have to mix together with any nitric oxide modulatory materials like hydrogel or polymer conjugates or proteins or micelles or polymeric coatings. So first off, my talk is about the stimulus response nitric oxide releasing systems. So in our lab, we are developing nitric oxide releasing piezoelectric nanoparticles for the deep brain stimulations. So in the case of Parkinson's disease, we are insert the electrode, and electrode can produce the electric current, and it can stimulate our neurons, and we can stimulate our neurons and releasing of dopamine for the treatment of Parkinson's disease. But that electrode insertion is very invasive, so we are delivering of the nanoparticles having the releasing nitric oxide for the opening of BBB, as well as producing electric current by stimulation of ultrasound. As you know, there is big barrier, brain-blood barrier in the brain. So this barrier can protect our brains from the foreign materials. But if we want to deliver any drugs or materials into the brain, we have to disrupt the BBB to deliver any drugs. So from the references, nitric oxide can open the BBB by the stimulation of MMP9 and by disruption of tight junction proteins. So we are using the nitric oxide for opening the BBB. So in general, the deep brain stimulation is very popular for the treatment of Parkinson's disease. In the case, we have to insert electrode to produce the electric current, and calcium voltage-gated ion channel will be activated, and some neurotransmitters like dopamine can be released from the neuronal cells and for communication of the neuronal cells. But this is very invasive treatment, so we are developing alternative to deep brain stimulations. We are using the piezoelectric material. If we stimulate it by ultrasound, it can produce electric current to stimulate neuronal cells. And also we are using nitric oxide releasing materials for opening the BBB. So we are using a BNN6. BNN6 can release nitric oxide when stimulated by ultrasound. So finally, we developed the nanoparticles inside the core, the BTMP bioimplanted nanoparticles. This is piezoelectric material, and we coated this nanoparticle with polydopamine for loading of BNN6 as anode releasing materials. So we injected this nanoparticle into the blood vessels, and it can circulate our body by via the blood circulations. And if we stimulate brain by the HIFU, you can stimulate this nanoparticle to release nitric oxide. Nitric oxide can open the BBB, and then these nanoparticles will go into the brain tissues. And if we further stimulate this nanoparticle with a HIFU, it will stimulate the nanoparticle to produce electric current to activate the neuronal cells. So firstly, we injected the nanoparticles into the blood vessels, into the tail vein of the mice, and then check the BBB opening. And then we, after injection of nanoparticle, then we stimulated the brain by a HIFU, and then we injected the staining reagent into the blood vessels, and then you can see the stain reagent are accumulated in our brain by stimulation of ultrasound, ultrasound and the BNN6. It means by the stimulation of ultrasound, this material release nitric oxide, and nitric oxide will open the BBB, and these materials go into the brain tissues. And we also check the barium concentration of the brain by the stimulation of ultrasound and nanoparticles. We can see we obtain higher concentration of barium in the brain tissues. And then we study the ultrasound-driven deep brain stimulation in Parkinson's disease. We injected nanoparticle into the tail vein and then stimulate by ultrasound, and then check the behavior test using the ROTA test and open field test. So in normal mice, it takes long time from the rotating route, because it is very normal. But in Parkinson's disease model, it's very quickly falling down from the rotating rota. Okay, so we can measure the time to falling down of the mice. This is normal mice. It takes a long time, but this is disease model. They falling down very quickly, and after treatment of the mice, its behavior is very similar to the normal mice. And then we check the the behavior of the mice. This is normal mice. They can move very actively, very normally. And all these disease model, the moving activity is very slow. This is treated mice. So the moving activity is almost similar to the normal mice. So it means by stimulation of ultrasound, you can open the BBB, and then you can stimulate the neuronal cells by ultrasound, and the Parkinson's disease behavior is recovered. So this slide shows the summary one of my talk. We developed piezoelectric nanoparticles releasing nitric oxide and producing electric current by stimulation of the HIFU. And by using these materials, Parkinson's disease model was significantly recovered. As I said, sometimes we have to remove nitric oxide because nitric oxide is an inflammatory material. So in our inflammation site, the nitric oxide concentration is very high, like autoimmune disease like IBD or rheumatoid arthritis or OA. The nitric oxide concentration is very high, even in the cancer cells. So by simply removing nitric oxide, we can treat the disease. So we are developing nitric oxide scavenging materials. At normal state, the activated macrophage can release nitric oxide to killing the bacteria or to kill the tumor cells. However, in physiological disorder statement, unknown factor will stimulate macrophage, and macrophage release nitric oxide, too much nitric oxide concentrations, and it can damage to tissues, and it can accelerate normal cell apoptosis. So in that case, we have to remove nitric oxide from the inflammation site. Like rheumatoid arthritis, there are a lot of inflammation disease, so there are a lot of concentration of nitric oxide in the rheumatoid arthritis. The nitric oxide can be upregulated, and it can damage to cartilage, and it can stimulate inflammation cytokines like TNF-alpha or IL-1 betas. So we have to develop any material, any nanoparticles or any other hydrogels for removing nitric oxide in the inflammation site. So we are focusing on the also phenyldiamine. This moiety has very high reactivity with nitric oxide to scavenge nitric oxide to form benzotriazole, which is very biocompatible materials. So we chemically conjugate, we synthesized nitric oxide cleavable cross-linker to form the polymeric hydrogel. This is nitric oxide responsive hydrogel. This is acrylamide, and this is nitric oxide cleavable cross-linker. For the polymerization of the acrylamide, we need acrylamide monomer, and we need bis-acrylamide for cross-linker. So for electrophoresis 40% acrylamide gel, this acrylamide will be polymerized to make the linear polymers. And by adding bis-acrylamide, the linear polymers can be cross-linked to form the gel. Instead of bis-acrylamide, we added nitric oxide cleavable cross-linkers. And you can cross-link the linear acrylamide polymers. And this moiety will react with the nitric oxide to form benzotriazole, and finally cleaved by hydrolysis. So this polymer gel network will be disrupted by reaction with nitric oxide. And we checked the nitric oxide responsive behavior, and then we activated the raw cell by adding the LPS, and it can produce nitric oxide. And then we added the hydrogel. This is normal cells cannot produce nitric oxide. There is no change in the gel networks, but this is will produce nitric oxide, and nitric oxide responsive gel was swelled by the disruption of polymer network disruptions. But this is control gel. There is no change in the swelling behaviors. Okay. So we size narrowed down into the nano-sized and injected into the rheumatoid arthritis site. This is nitric oxide scavenging gel. It can be injected into the site, and then you will remove nitric oxide from the RA site. So we check the inflammation cytokine like IL-6 and TNF-alpha by using nitric oxide scavenging gel. Inflammation cytokine level is recovered into the normal baselines. And then we check the rheumatoid arthritis score. If the inflammation is severe, the color will be red and very swelled. Okay? But as you can see, the score was recovered to the normal. This is nitric oxide scavenging gel, removing gel. This is dexamethasone as a positive control, and this is nitric oxide non-scavenging gel. Okay, there is no efficiency in for the treatment of rheumatoid arthritis, but in nitric oxide scavenging gel, we can see considerable treatment efficacy in rheumatoid arthritis. So for the advancing system, we developed two solution system. This is one solution having the hyaluronic acid, the azide modified hyaluronic acid, mixing with nitric oxide cleavable of cross-linkers. And the other solution has micelles having the azide bonding. So by mixing together, they can quickly react to each other to form the gel. And dexamethasone was loaded into the micelles. They will form the gel, and by the reaction with nitric oxide, the nitric oxide cleavable cross-linker will be cleaved. So the micelle will be released from the gel, and finally, the dexamethasone loaded inside the micelle will be released from the micelles. So by using this system, we can treat rheumatoid arthritis by using the dexamethasone as well as the removing nitric oxide from the gel. This is normal gel. We can mix together one solution with another solution. We can see very simple polymer hydrogel. But we can inject together by using the dual injection system. We can quickly see the very stable polymer hydrogel. This gel is maintained a very long time inside the mice. And then we check the therapeutic effect of micelle nitric oxide gel in vivo. We injected collagen for the RA model, and then we injected this polymorphic nanoparticle gel into the injections into the articular site, and then checked the efficacy. If the normal mice has very dense bone density, but the disease model, the bone density was very low. However, after treatment of the mice with our gel, the bone density was recovered. The bone density is very high, and other inflammation cytokines recovered to the normal state. So this slide shows the summary of the theme 2. We injected injectable the solution to form the instantaneous hydrogel to removing nitric oxide and release dexamethasone to for the treatment of RA. So as I said before, in our lab we have the startup company licensed out from our university. So we are developing our nitric oxide scavenging system for the clinical trials. So as I said, in physiological condition, the inflammation site has very higher concentration of nitric oxide. Okay, so nitric oxide can be produced from the INOS, the enzyme. There are three kinds of NOS, nitric oxide synthase, ENOS and INOS and NNOS. So the most problematic NOS is the INOS. So in inflammation site, INOS will react with arginine to produce high concentration of nitric oxide. Okay? Also, so we can see very higher concentration of nitric oxide in the cancer cells. So this slide shows the level of nitric oxide so we can see in RA model, the nitric oxide concentration is very high compared to our other normal controls. And also in IBD model, so nitric oxide also very high. So for the treatment of IBD, we have to remove nitric oxide, inflammatory bowel disease. And also in colorectal cancer, nitric oxide concentration is very high, and other carcinomas, nitric oxide concentration is very high. So we have to remove nitric oxide from the cancer cells and from the autoimmune disease site. Previously, the Pfizer developed nitric oxide inhibition drugs, but in phase 3 clinical trials, finally failed because the efficacy was not so good. They used INOS inhibitor as a drugs, but we are using nitric oxide scavenger for the treatment of this autoimmune disease. We can use nitric oxide scavenger itself, but we are chemically conjugated nitric oxide scavenger to the FDA-approved drugs. Okay, so this is FDA-approved drugs, and we chemically conjugate nitric oxide scavenger into the drugs. So in this state, it cannot bind to the target site. So it means it cannot show any drug efficiency. However, at inflammation site, this linker will be cleaved by nitric oxide at the autoimmune disease site or cancer cells. Finally, this nitric oxide drug conjugate will go into the original drugs, and original drugs will show the drug efficacy. We developed this system, nitric oxide scavenging drug conjugate, like antibody drug conjugate or small molecule drug conjugate. The concept is very similar to the ADC, and it can be it has a potential in autoimmune disease, and it can be targeted to the inflammation cells or inflammation tissues. Okay, and also we can develop it as a oral treatment. In autoimmune disease like IBD or RA, JAK inhibitors like tofacitinib or other drugs are very popular and is very blockbuster drugs. However, it has very high toxicity, so we have to be very careful to use JAK inhibitors for the treatment of IBD or other autoimmune disease. Okay, so this NSDC concept based on the tofacitinib will alleviate toxicity issues of JAK inhibitors. So, as I said, in the normal tissues, the nitric oxide scavenger still chemically stably conjugated to the JAK inhibitor drugs. However, at inflammation disease site will be cleaved, and these drugs will go into the original drugs, and this will activated. So there are three kinds of JAK inhibitors in the clinics: the tofacitinib and the baricitinib and upadacitinib. All these drugs are very blockbuster drugs and very popular, and the sales is very high, 2.4 billions and 1.1 millions. But all these drugs are very toxic, so we have to conjugate any chemicals. In RA system, MTX is the first drugs, and then the UC, ulcerative colitis, the biologics like anti-TNF-alpha or the JAK inhibitors are very popular. However, the biologics has some tolerances after 2 or 3 years later, and the JAK inhibitors has very high toxicities. So we chemically conjugated NO scavengers to the tofacitinib. Sorry for not showing the detailed structures because of the patent issues. And we conjugate by reaction with the nitric oxide. This link will be cleaved, and this drug will go into the original drugs. So we don't for the clinical trial, we don't have to do using the tofacitinib, because tofacitinib is already FDA approved drugs. So we can just only using any we can just prepare any documents for this material. Okay, so firstly we check the efficiency of nitric oxide scavenger itself, not conjugated with the tofacitinib. In RA model, just simple treatment of using the nitric oxide scavenger, we can see very high efficiency in arthritis scores. And also in vivo UC model, we also obtained very high efficiency by using just simple nitric oxide scavengers. And then we prepared the in vivo UC model, very acute models, and you can see we treated this mouse using our NSDC model, nitric oxide drug conjugate. This is very severe model, and this is tofacitinib as a positive control, and this is our drugs. Compared to the positive control of tofacitinib, our drug showed very high efficacy in DSS UC model. This is the normal length of colon length at the normal mice, and this is disease model, DSS model, and these are our treated models. So colon length was recovered into very similar to the normal. Okay, so by using our nitric oxide scavenging drug conjugate, the colon length significantly recovered into the normal. And we also developed the semi-UC model. Compared to the tofacitinib as a positive control, we also obtained a very positive result of our candidate nitric oxide drug conjugate. This is data from Charles River in DSS UC model, and you can see very similar efficacy compared to the data from the Charles River, the one of very famous CRO in the world. So this is summary of my talk. We developed nitric oxide scavenger conjugated drugs. At the normal stage, it cannot show any drug activity, but in inflammation disease site, it can be cleaved and the drug will be activated again. So we can, this is platform technology. We can apply it for any drugs like topisteam or anti-cancer drugs or other drugs having very high toxicity. This is end of my talk, and I want to say many thanks to the audience, and thank you for your attention.
Thank you very much, Professor Won-jung Kim, for your very impressive talk with many interesting pieces. So, any questions from the floor first? I have a very simple questions. So, as far as I know, NO is a kind of a gas molecule. So, it's a kind of very unstable molecule, right? So, I'm wondering, how do you keep the NO-loaded material stable until kind of a animal study or in vitro assay?
Yeah, very good question. Nitric oxide itself is very unstable molecules, but if it conjugated to the NO donor, it is very stable. So in donor stage, the nitric oxide cannot be released from the donor. It's very stable.
Okay. Thank you, Professor Won-jung Kim, for your talk.
Thank you again.
So we are moving on to the next speaker. The CEO, Sin-gyu Bae, is a founder and CEO in MD Immune Incorporation. So he's talking about Biodrone as a smart drug delivery platform based on cell-derived vesicles. Please join me welcome CEO Sin-gyu Bae.
Thanks for the kind introduction. I'm Sin-gyu Bae. I'm the founder and CEO of MDimmune. MDimmune is a company established with the goal of developing innovative therapeutics that can provide hope to the patients suffering from incurable disease due to the lack of appropriate therapies. To achieve this dream, we are developing a next-generation smart drug delivery technology called Biodrone platform, which can safely deliver drugs to targeted tissue. Drug delivery technology is a crucial aspect of drug development and has been under development for a long time. However, existing drug delivery technologies still face challenges in terms of safety issue and the efficiency of drug delivery into the cell, and particularly in delivering drugs to intracellular targets. The Biodrone technology based on cell-derived nanoparticles represents a next-generation drug delivery system that can address the limitation of traditional drug delivery systems. Cell-derived nanoparticles are biocompatible and exceptionally safe. Moreover, it efficiently undergoes uptake into target cells, allowing simultaneous delivery of various drug modalities, such as protein, peptide, and nucleic acid. This technology holds the potential to overcome the challenges associated with conventional drug delivery systems. The Biodrone platform is created through a cell-to-nanoparticle conversion process using an extrusion technology. As you can see, it generates tens of thousands nanoparticles from a single cell. The extrusion process utilizes a membrane filter and employs a serial extrusion for efficient nanoparticle production. This video clip shows the cell extrusion process through 3D animation. The cell, with its lipid bilayer, is fragmented into appropriate 100 to 200 nanometer sized particles. When passed through the membrane filter under pressure, the resulting nanoparticles closely resemble extracellular vesicles in size and characteristics. The nanoparticles produced by external process are we call cell-derived vesicles, CDVs. Their exceptional characteristics originate from their parent cells, ensuring high biocompatibility. Additionally, CDVs can encapsulate a diverse range of drugs within their nanoparticles and can be precisely targeted to specific tissues. These characteristics allow for the enhancement of their functions through the engineering of CDVs. CDVs nano-sized vesicles featuring lipid bilayer membranes similar to extracellular vesicles were systematically examined. Morphological analysis using cryo-TEM and SEM were conducted on CDVs and extracellular vesicles derived from UCMSC and HEC293 cells. Various physical properties, including size distribution, polydispersity index, zeta potential, etc., were studied. Notably, CDVs exhibit physical properties that are highly similar to those of extracellular vesicles. Although CDV exhibits similar physical properties to extracellular vesicles, cellular uptake analysis revealed CDVs demonstrate enhanced cellular uptake compared to extracellular vesicles. Experiment data on several target cell lines have shown that CDVs have about twice the cellular uptake efficiency compared to extracellular vesicles. As I mentioned regarding the superior safety profile of CDVs over existing drug delivery vehicles, T-cell proliferation assays confirm CDVs have quite low immunogenicity. In vivo safety analysis of CDVs conducted through GLP-compliant toxicology studies have confirmed their safety. Comprehensive toxicological assessment, including repeated dose toxicity, immunotoxicity, and genotoxicity, were performed. With natural killer cell-derived vesicles administered over 4 weeks, no detectable toxicity associated with the CDV administration was observed. Notably, CDVs exhibit markedly lower immunogenicity compared to lipid nanoparticle. We administered 0.3 mpk mRNA via intravenous route using both LNP and HEC293 CDV. Subsequent analysis of serum cytokine levels at 6 to 48 hours post-injection revealed a substantial elevation in pro-inflammatory cytokines following LNP injection. However, no such changes were observed in the case of CDVs. This highlights the favorable immunogenic profile of CDVs in comparison to lipid nanoparticles. Biodrone is targeting two significant markets. The nanoparticles produced by extruding stem cells and immune cells can enter the next generation cell therapy market. Additionally, by utilizing HEC293 cells and RBC, etc., Biodrone can enter the market for drug delivery systems that deliver proteins and nucleic acid. Both of these markets are substantial and currently experiencing high growth rate in the global market. Our first core technology is the cell extrusion process, and the second core technology is CDV engineering, as you can see here. While CDV possess tissue targeting capabilities based on parent cells characteristics. However, that's not good enough. Through CDB engineering, we need to enhance targeting and drug loading functionalities further. To do efficient CDB engineering, we identified 4 highly expressed CDB membrane anchor proteins. By attaching targeting ligands to the extracellular domain of these anchor proteins, we can further enhance targeting efficiency more and more. For example, linking nanobody with a strong affinity for the transferrin receptor to CDBs resulted in a remarkable 36-fold improvement in brain delivery. I will elaborate on the research on identifying the 4 membrane anchor proteins, which are the core of the CDB engineering technology. We first analyzed crucial surface markers of HEC293 CDBs and EVs, revealing notable differences. LMP1, NCSTN, and CD63 were more highly expressed on CDBs, while CD81, CD9, and PTGFRN were elevated in extracellular vesicles. This further demonstrates the enrichment of subcellular organelle membranes alongside plasma membrane components and reveals a distinct vesicle biogenesis compared to extracellular vesicles. Therefore, we conducted whole proteome profiling to identify membrane protein specific to CDBs. Although specific membrane proteins were overexpressed in cells, it was not certain that they would be abundantly present in the resulting CDBs. To facilitate robust CDB engineering, the identification of stable specific membrane anchor proteins were essential. HEC293 cell CDB proteome analysis revealed multiple candidate anchor proteins that are relatively abundant in HEC293 CDBs compared to HEC293 EVs and parent cells. Through the proteomic study, we have identified 4 distinct CDB anchor proteins and subsequently filed a patent for this engineering technology. Anchor protein A and B primarily localized to the plasma membrane, while anchor protein L and P exhibit localization to the endo or lysosomal membranes. Through CDB engineering technology, we have significantly enhanced the tissue targeting capabilities of CDBs. We show an example in central nervous system targeting research. The binding of nanobody to a transferrin receptor on the outer surface of anchor protein results in a remarkable 36-fold improvement in brain targeting. Therefore, it has been confirmed that the CDB engineering technology based on membrane anchor protein is working great. Through this approach, it becomes possible to deliver CDBs to the desired tissues within the body. CDB engineering technology not only enhances tissue targeting capabilities, but also facilitates efficient loading of protein drugs. Fusion of GFP with membrane anchor B yielded a noteworthy 76% of GFP positive vesicles assessed by a nano-FCM instrument, in contrast to the 13% observed in the case of cytosolic expression. Anchor engineering allows for the loading of protein cargo up to approximately 350 molecules per single vesicles, depending on the protein concentration within the cell. Recently, with the success of COVID-19 vaccine development, gene therapy has gained significant attention. In the development of gene therapy, the use of drug delivery system is essential. We are conducting research on loading genes through both exogenous and endogenous methods. Small-sized RNA is effectively loaded using the exogenous loading approach, while for larger-sized mRNA, we are exploring the endogenous loading method. Therefore, by effectively combining CDMO engineering technology with the exogenous loading approach, we can load and deliver both siRNA and proteins simultaneously within a single CDB. When delivering drugs into cells, the efficiency of endosomal escape after cellular endocytosis is crucial. We are able to increase endosomal release sixfold by using suitable enhancers. Typically, the endosomal release efficiency of LNP drug delivery system is reported to be less than 1% or approximately 1 to 2%. We are actively engaged in multiple projects involving the delivery of RNA therapeutics, proteins, and other drug modalities. We are collaborating with the Australian National University on a research project focused on incorporating microRNA to address dry AMD. Additionally, we are conducting research to assess the therapeutic efficacy of the simultaneously loading siRNA and peptide drug together for inflammatory and autoimmune disease. Furthermore, we are conducting research on delivering RNA to the brain, demonstrating significant potential for addressing unmet needs in CNS. This area research has garnered substantial interest from major international companies. Dry AMD therapeutics development program with the ANU is one of our main projects. You can see the in vivo data here. The microRNA 1 to 4 loaded CDVs demonstrate an improvement in retinal function, concurrently mitigating cell death and retinal damage. CMC is very important for the therapeutics development of cell-derived vesicles as well as extracellular vesicles. We have established manufacturing process, including cell extrusion process and vesicle purification process, such as TFF and size exclusion chromatography, ensuring batch consistency. Furthermore, our CDV manufacturing process exhibits high productivity compared to EV, about 10-fold to 100-fold, demonstrating commercial competitiveness. Our Biodrone platform technology can advance step by step. The first generation technology enables therapeutic development just only through the cell extrusion process. Very simple process without any vesicle engineering and drug loading. And second generation technology enhances more therapeutic efficacy through additional drug loading process with endogenous and exogenous loading method. Third generation technology combines drug loading with specific targeting functionality. This will be our final product to develop the best Biodrone platform. So far, we have signed 3 small licensing out deal agreement and also have a collaborative relationship through networking with global companies such as Lonza and J&J. Our company has consistently pursued research and development so far. Since 2020, we have engaged in collaborative research with not only domestic companies but also global partners to make synergy effect. Global pharma and biotech companies have high interest in Biodrone technology as seeking advanced drug delivery solutions. This reflects a high level of international recognition for Biodrone technology. Actually, our business model is to provide smart drug delivery solution to the partner companies. MDMN was established in 2015 and is commercializing biodrone technology. We have received around $30 million in investment so far, and we have a local subsidiary, Biodrone Therapeutics, based in Seattle, USA. Additionally, we have built a strong patent wall with 18 active patent families. The leadership of MDMN consists of 4 executives with extensive experience in research and business development. This is the entrance of our lab. You can see the phrase about why we need to do this research. We are engaged in the work of saving lives. So we have to do this work, this research, and this business. Someday, I am confident that this dream will come true. Thank you for your attention.
Thank you so much for your impressive talk. So, questions from floor first. I have a question. So you are discriminating your CDV technologies from EV. So I'm wondering what is the most distinct difference between your CDVs and extracellular vesicles?
Yeah, a couple of differences. First is productivity per single cell, and our extrusion process enhance the productivity 10x to 100x, depending on the cell types. And the second is we can use just a cell itself. I mean, for the EV production, we need to culture the cell, right? But we can use the extrusion process with just the cell. I mean, so we can make the CDBs with the red blood cell or platelet. They cannot difficult to make the EVs from the cells because they don't have nuclear, and so they can not proliferate, right? So, yeah, and so this the engineering process is membrane. Now, surface membrane proteins are a little different, so we can find the specific membrane proteins to CDVs. It's not abundant in EVs, and then we can use use that anchor proteins. Yeah.
Okay, thank you for the question, the answer. Actually, I have many technical questions, but limited of time. So I'm wondering just one quick question.
Yeah.
So you took endogenous messenger RNA loading strategy in order to load messenger RNA into your CDVs. So would you be more specific on what's the endogenous messenger RNA loading strategies?
It's based on the cell engineering technology, and then the main idea is the cell produce messenger RNA inside the cell cytosol, and then bind to RNA binding proteins specific to the membrane anchor proteins.
Oh, very interesting. Yeah, thank you. So it was great talk. Thank you very much for your wonderful talk.
Thanks again.
Yeah, we are closing the session. So next session will be open on 3:30 p.m. at this hall. Thank you. Thank you for coming today.
information technology and security, customer service, and operations. Prior to joining Samsung Biologics in 2014, he was CIO at Beckman Coulter's Clinical Diagnostics and Life Science Businesses, a CIO and CTO at Integrity, and a Senior Director of Information Systems, Customer Service, E-Business Technology, and Site Planning at Philips Healthcare. He holds a bachelor's degree in mechanical engineering from the University of California, Irvine, and an MBA from the University of Southern California. Please welcome, give a big applause to him.
Thank you. Can everybody hear me okay? Hi, thank you for allowing me to speak at this conference. Today I'm going to talk about keys to success in a challenging global CDMO landscape. CDMO, I'm assuming everybody understands, stands for Contract Development and Manufacturing Organization, which is what Samsung Biologics is. So I'll give you a little bit of overview of the industry and our business and what we're doing to invest preemptively to continue to be successful in this industry. And then we'll do a Q&A at the end. First, let's talk about the growth drivers, the macro things that are happening across the globe. As everybody knows, as especially we here in Korea are well aware, people are getting older. The population is getting older. I think in Korea alone, the projection is that by the year 2025, there'll be people over the age of 65 will be 20% of the population or more. On the other hand, people are living longer. The average age, the lifespan is going to go up to 85. That's significantly higher than the OECD average of 83. So Korea is doing a lot of great things to prolong the lives of people, but on average, the age is getting older, which means greater healthcare needs in the future. Second macro indicator is higher prevalence of diseases. We all felt the impact of the global pandemic. Nobody saw COVID-19 happening. But before that, we had the bird flu. We had the H1N1. We had the Middle Eastern virus. These things constantly happen. We're seeing these things happen, and we're going to expect to see these diseases continue to take over. But on the flip side, there's technology advancements. There's more treatments that are being developed every year to counter these diseases. And this is where the last point, and as a result, the industry itself is addressing the need, especially with CDMOs, by making these treatments more accessible, making them more affordable, scaling up production so that more people can get access to it. Quick explanation on biologics. It's in our company name, Samsung Biologics. What are biologics? Well, biologics are a type of drug compared to chemical drugs. Chemical drugs, are like aspirin, things that you typically swallow in a pill format. Very simple molecules. Biologics are derived from living organisms. Okay? Biologics, especially antibodies, are adapted. They take advantage of the human body's own adaptive immune system. So they're more targeted, they're more effective because it uses a natural way to fight cancer and other types of diseases like autoimmune diseases. So as a result, biologics has seen a huge growth compared to chemical drugs. Chemical drugs, compound annual growth rate, CAGR, is about 6.9%. Okay? Biologics, on the other hand, is estimated to grow at the single double digits, 10% or more. Okay? And this is very exciting because not only are these drugs more promising, but there's going to be more of it, more industries are going to go after it. Okay? So speaking of industries, the question is there's there's that growing demand for biologics. Will there be enough supply, enough manufacturing capacity to supply that demand? The forecast, the current forecast, several different ways to look at it, says no. Not by the year 27. There's going to be a shortfall. We hear about plants, people investing in plants, companies investing in growth. But what you don't hear about are companies that have aging facilities that are now coming off production. Okay? So overall, there's going to be a greater demand and there's going to be a shortfall in demand. This is why Samsung Biologics, as I'll explain later, is continuing to invest in our biologics manufacturing capacity. Okay? CDMOs, the commercial, the contract manufacturing development is seeing very healthy business growth as a result, 13% compound annual growth rate. But what's driving this growth? We talked about the macro factors behind the overall healthcare industry. But specific to biologics, what we're seeing recently is a rise in GLP-1, obesity drugs, right? How many of you guys heard about Wegovy? Wegovy. Elon Musk said, he went on Twitter and said he lost his weight by taking Wegovy, right? It's a very promising weight loss drug. Eli Lilly is also working on a type of drug to similarly allow people to lose weight. There's a greater prevalence of multiple indications. So whatever the drug was designed to treat, now it can treat other types of indications and diseases. Or there's combination therapies. So the same drug now suits multiple different indications. Okay? We're seeing that with Merck's drug, Novo Nordisk. Again, going back to Wegovy, not only does it treat weight loss, but it also helps to treat diabetes. So we're seeing that with other manufacturers that, the original indication, now the drug can also attack other types of treatments. We see a boom in biosimilars because more and more drugs are slowly coming off patent. And biosimilars, for those of you not familiar, are kind of like the generic equivalent of biologic drugs. Chemical drugs can have the exact same chemical composition, so it would be the exact same molecule structure. Biologics, on the other hand, are a little different. They're more complex, so they're not an exact duplicate, but they're similar in terms of the antigens and the antibodies and the whole makeup. So we're seeing more and more developers, more and more drug companies pursuing biosimilars, which is also driving demand. And the last one is potential blockbusters. Alzheimer's. Alzheimer's, if these drugs get approved, and they're on the way to getting approved, if they're successful, can really drive tremendous demand for manufacturing capacity. Okay? So CDMOs, as a result, are responding by first building out and making sure we have enough capacity, but also speed. Speed to deliver those capacities to market so that the manufacturers can deliver their drugs to market faster. And then third is innovation. And Samsung Biologics is proud to say that we've addressed all of these three things. It's been part of our core strategy from day one, right? Starting from plant one, we continue to build out plants. We've now completed our first biocampus and are constructing our second biocampus. And as a result, we've accumulated the world's largest total mammalian drug substance manufacturing capacity. That's 604,000 liters. We also not only have the largest total capacity, we've also built them 40% faster than the industry average. We could do that because we could leverage the expertise that we have as a Samsung group of families, Samsung family of companies like Samsung Engineering, Construction. We can do these things much faster because we have that expertise in clean room design, hygienic piping, large petrochemical type facilities. We can do that. So we leverage that expertise to build these things 40% faster. Operationally, when we win a contract, we have to do a tech transfer, basically following the SOP and the way to scale up that drug to our large-scale manufacturing capacity. Normally it takes like 12 to 18 months to do that. We can do that in half the time or less. In fact, at the height of COVID, we were able to transfer one of our clients' drugs in 3 months as opposed to 12 months. And the third thing is innovation, right? If you look at the D in CDMO, development is what leads to manufacturing, commercialized drugs. So to attract our development clients and to make sure that they have the right tools, that they have the tools necessary to develop those drugs, we've come up with our proprietary platforms, including DevelopPick, S-Chosient, S-Dual, dual antibody, S-Choice, which is our cell line platform based on the CHO cell, and S-Glin. But beyond monoclonal antibodies, which is a lion's share of our business, we're looking towards the future and investing in mRNA. We saw how successful mRNA was in being able to deliver vaccines much faster during the pandemic. And the next wave, the next generation, if you will, of antibodies is antibody-drug conjugates, where you now have the antibodies with the cytotoxins that could kill the cancer cells. Okay, so we're investing in these technologies to be able to provide services to the market. Total business, all of this has led to our business performance. We now have over 110 clients, many of whom are global big pharma. In fact, if you look at the top 20 largest global pharma companies in the world, 14 of them are our clients. We're very proud of that. We've exceeded $3 trillion last year in total sales. And for 10 consecutive years, our clients voted us as one of the top CDMOs, okay? And the reason is that not only can we build plants fast and provide the capacity, but we also have the ISO certifications to prove our operational excellence. The three that you see highlighted there with the star and the red, these are very important to our clients because we need to protect our clients' intellectual property. They need that assurance and that's, the safety and the assurance to know that their service provider is protecting their intellectual property. All this, as I said, we've had some phenomenal, unprecedented business growth. We started out, back 12 years ago, but now over the years, we've had a very successful growth rate and got to 3 trillion Korean won last year. We did it all out of here. Incheon, Songdo, just down the street. It took me less than 5 minutes to get here from our office, right? We want to thank the city of Incheon, the Songdo IFES district for making that possible. A lot of our clients that come visit us say, why Songdo? And we say, why not Songdo? It's central to Europe, it's central to the United States. We export out of here to all these different countries that our clients market their products to. And as a result, over time, Songdo has now become like an economic hub. It's become an ecosystem for biotech manufacturing, right? So not only in Korea, but probably in Asia, with some of these largest, these core suppliers that all the CDMOs and pharma companies all use, Sartorius, General Electric, Cytiva, Saint-Gobain, all the ones you see there. Some of them are actually starting to construct manufacturing operations right here to provide their services directly from Songdo. As I mentioned before, Biocampus 1 is completed, and we are really excited about Biocampus 2. We've actually started construction of our Plant 5, which will go online by 2025, within 2 years of groundbreaking, unheard of in this industry, okay? So I talked about capacity, I talked about speed, innovation, but there's a fourth factor that companies need to invest in, and that's people. We've been using this tagline, driven for life. We've introduced this campaign about 2 years ago. It makes sense to us because driven for life really symbolizes Samsung Biologics. Driven symbolizes Samsung. Samsung is driven for solutions. We're innovative. We don't stop until we find solutions. For life, that's what biologics is. So driven for life is a great way to represent Samsung Biologics to our clients. But we've been using this campaign as a theme to say we're also driven for people. You can't run a business this successfully without people. Okay? I said we grew from 12 years ago to where we are now, and we have about 4,500 employees. We've had to recruit an awful lot of employees, a tremendous amount of employees. As a result, our average age is about 30. It's a very young campus compared to a typical biopharma company like us. Our clients always visit us and they're surprised. Wow, you guys are like a young college campus. How do you do it? Well, explain how we do it. But one of the things that we had to address is not only is our talent force, our workforce, very young compared to other pharma companies, we also have a very diverse ratio. Okay? 40% of our employees are females. That's very, very different than typical biopharma companies. Okay? As a result, we've invested in professional development. We've had a more horizontal leadership structure. Our CEO, John Rim, directly engages through town halls and sessions. All of our leadership conduct town halls within the center. How do we invest in our employees? Well, this is one way we do it. We're sure that many people in the industry do things that are similar. One is we train and educate. In this area, we leverage the Samsung innovation on technologies like virtual reality, augmented reality. We actually used VR and augmented reality during the pandemic. We invested in that because when our clients and the FDA and the EMA, they couldn't travel to our site to inspect our facilities, we used VR to help them virtually inspect our facilities. And we gained approvals at the height of the COVID pandemic. But we're now leveraging that technology so that using a digital twin of our environment, of our manufacturing environment, new employees can actually virtually practice operating the equipment before they actually gown up and go into the facility. Second is sharing our vision through mission and pride. We've had a campaign. 30-year-olds, they don't know about getting sick. They're still healthy. So we have to give them a sense of mission and say, you may be healthy, but your parents, your friends, the average person in your neighborhood, right? They may need these drugs that you're making. So we've invested in a program, in a campaign to internally promote awareness and build that sense of mission and pride in the work that we do. I mentioned our employees are 30-year-olds. I think the Korean term is millennials or MZ generation, right? The way you connect with them, the way you engage them has to be different. So we actually have a reverse mentoring program where people like me get mentored by new hires, people that have been on the job for less than a year, right? They tell me how to get on social media, how to connect better, right, with the younger workforce. And last is engaged. We got to keep our employees interested, motivated, give them the benefits needed to really motivate them to come to work every day. We invested in BioPlaza 2, and it is really state-of-the-art. I'm not going to go into all the things we have, but it's got its own clinic, it's got its own dentist office, it's got Starbucks, it's got Olive Young, it's got a cafeteria, it's got an educational facility that could lecture over 1,000 people at once. It's a tremendous benefit that we provide, and it really allows our employees to, especially our employees who have to, travel from long distances or their homes. Their hometown may be hours away in a different part of Korea. They live in the dormitories. They get access to these facilities to make sure that they're taken care of. So I'd like to close with a policy recommendation, and that is if you look at how much workforce Korea, let's just look at Korea, is going to need by the year 2027, when the average lifespan is 85, and people 65 years or older is 1/5 of the population. We're going to need a workforce of over 100,000. And right now, the best projections say it's going to be much less than that. Huge shortfall. Huge shortfall. 75,000 is what the projection is, based on our current growth, population growth. So we have to invest in talent. As a nation, we need to invest in talent. And there's some examples from other countries, like Ireland has the National Institute of Bioresearch and Technology, where the government of Ireland has invested in promoting biotechnology and the critical skills needed to have more people that are capable of joining the biotech workforce. China has a program to find people with Chinese ethnicities with foreign PhDs and bring them back to China. They're doing a pretty good job at it. They've got a lot more PhDs in Korea. They have a lot bigger population, but Korea should really cherish its advanced degrees, people with advanced degrees, and find ways to attract them back to Korea. And lastly, we need biotech clusters like IFAS, like what we have in Songdo. Continue to invest in biotech clusters because that'll create, and attract talent, not just within Korea, but global talent to centralize and really help nurture that workforce. So in closing, it's not just about facilities, not just about technology and equipment, but it's also about talent. The talent needed to operate and run these GMP facilities. And it's really the engine. People are the growth engine, okay, for innovation, excellence, sustainable growth. And this is going to be a collaborative. It's going to require the collaboration of industry, government, and academia to get together and really promote and grow the talent that we need for the future. Thank you.
Thank you for your talk. Among the audience, do you have any questions?
I have a question.
Oh, yeah.
Actually, you already mentioned some policy recommendations, including education and collaboration between government and your company. But what do you think is the most important element to stimulate the ecosystem for CDMO business in Korea? And I have one more question. Is there any policy you think which should be, which the Korean government should focus on for CDMO industry?
I think the Korean government has been very helpful overall. Samsung Biologics would not be here, would not see the success that we've had today without the help of the Korean government, without the city of Incheon. So we've been very, very appreciative and thankful for that. So our position is that they continue to foster the industry as it has been. Our focus here today was the government can only do so much. It's up to the industry to work with also the academia to really attract and grow the talent needed, especially for the biotech workforce that we need for the future. So again, we're very thankful for the Korean government and the city of Incheon for all of the success that we've had so far. Thank you.
Thank you. I think you said that the most important element is the workforce for the ecosystem. I see.
Yes.
You have the mic over there.
You can use the mask, the mic at the back of you.
Yeah, thank you very much for your presentation. My question is, I think in Songdo is an area of hub for biotechnology, but what is lacking is more of a small and medium company. So as a large and global company, do you have any program that you collaborate together with small and medium companies and bring some synergy together
Yes, thank you for your question. One of the things that I did not mention, especially as it relates to the future and our future strategic planning, is one of the ways that we're investing in smaller startups is through our life sciences fund. And you may have seen in the press recently, we've invested in a Korean firm called Araris to help us develop ADC-type platforms. So we continue to use that vehicle for venture capital funding to help small biotechs, not just in Korea, but also globally. The other thing I'll mention is that in our BioCampus 2, ultimately, when it's finally completed, one of the things that we're considering is an open innovation hub so that it can also foster those types of startups and collaboration. Thank you.
As she asked a question, actually, under open innovation environment, I think the role of Samsung will be very important. Yeah. Do you have any other questions? Okay. Thank you for your presentation. Thank you. Okay. I will introduce next speaker, Dr. Just a minute. Dr. Hee-Kwon Kyung. He joined Korea Institute for Industrial Economics and Trade in 2019, right after his postdoctoral education at Michigan State University. His research interest is focused on semiconductor, biopharmaceuticals, and dynamics between international politics and global value chain. As a successful policy analyst and consultant, mainly serving ROK Ministry, Hee-Kwon is now more than ever convinced that a biopharmaceutical is the key for national economy in the years to come. And so what he'd like to talk about today is a biopharmaceutical industry through the lens of geopolitics and economics. Okay, please welcome.
Distinguished guests, thank you very much again for coming all the way down here, Songdo, Incheon. It's far from your country, your home, and I see a lot of you are exhausted already. So I'll make my presentation as interesting as it could get and short. I'm Hee-Kwon Kyung, an Associate Research Fellow at KIET, Korea Institute for Industrial Economics and Trade. I did my doctoral study in economics at Michigan State University, and now helping shape Korean government's policies for industries of strategic value and economic security. The keywords of the time we are currently living in, as you all know. Today, I want to frame my talk or presentation based on the remarkable similarities or resemblance between semiconductors and biopharmaceuticals, namely chips and shots, in multiple dimensions, and explain the reason why Korean government and the major corporations are dead serious about making biopharmaceuticals as the next semiconductor industry. Thus convincing you that we are really committed to fostering a vibrant and dynamic ecosystem right down here, Songdo. And finally, why you can be sure that your business here will be successful. So now, these days, the public in general are very much interested, or if I exaggerate a little bit, obsessed with artificial intelligence and semiconductors. The microelectronics revolution, come to think of it, has been 80 years or something. Unlike the current enthusiasm for companies like NVIDIA, Advanced Micro Devices, Apple Qualcomm. For those of you in your 20s, young people, it was all about Intel back in the days, from mid-80s to early 2000s. When Intel first released their much acclaimed x86 microprocessors in 1979 to overcome and pivot away from the DRAM market, based on which Intel was founded because of the cutthroat competition from Japanese companies like Toshiba, NEC, and Hitachi. Nobody knew what Intel's x86 processors could do and how far the implications of personal computer could reach. By the way, my job at work is mainly about semiconductor industry. Been studying the chip industry like 5 years now. But let me say to you as a friend and a fellow student in emerging bio industry, I think I am watching a dormant revolution and a tremendous potential finally manifesting themselves. Since Watson and Crick's pioneering research and DNA sequencing techniques, synthetic biology powered by billions of transistors is about to bring about, I'll say, a whole new world. And I'm not the only one. This is the title of McKinsey report, The Bio Revolution. Let me give you some numbers. Since iPhone, now the annual smartphone shipments, the quantity of smartphones manufactured in a year is about 1.3 billion, with an average selling price ranging from $300 to $400 per device. What if some miracle cure comes out and every single one of the people take a shot annually? For example, let's suppose a shot that has to be taken on an annual basis, and if you take the shot, you'll live 20 years longer. But costs are about $10,000. With population rapidly aging at an unprecedented speed, I'm telling you that the mobile revolution and the market size it created for billions of people will be very small compared to the next miracle unleashed by innovative new biopharma products. Now, let me be more specific about the similarities between chips and shots, going back to about 80 years, some history. The transistor technology was developed around 1950 by Dr. William Shockley, John Bardeen, and Walter Bratton at AT&T Bell Laboratory to minimize the landline telephone operation costs. And the discovery of the double helix structure of DNA was also around 1950, for which Crick, Watson, and Wilkins got their Nobel Prize in 1962. Dr. Shockley, who was infamous for his bad temper, recruited the best masters and doctoral students when he opened up Shockley Semiconductor Laboratory at Mountain View, San Francisco. But the famous traitorous eight, the 8 men you're seeing here, escaped from Shockley Semiconductor Lab and found Fairchild Semiconductor. You see the F here? The name Fairchild came from one of the co-founders of IBM International Business Machines, George Winthrop Fairchild. So fed up with Dr. Shockley's management style, the traitorous eight asked one of them, Eugene Kleiner, the man with the number 2 on the picture, whose father was an investment banker, to find an expert who could invest and create a semiconductor company. Eugene's father recommended the legendary venture investor, later the first chairman of Intel Corporation, Arthur Rock. Arthur Rock, so impressed with these 8 men, and after about 30 or 40 investors turned it down the offer, finally found an investor. His name was Sherman Fairchild, who was the only son of George Winthrop Fairchild, and who in his early age inherited so much of IBM stocks. So the name Fairchild Semiconductor, and it wasn't really a new and independent company. by the Department of Fairchild, Cameron. In fact, the traitors 8 became employees again after a desperate attempt to get out of Shockley lab. But Robert Noyce and Gordon Moore, the man with the number 1 and 3, and by the way, Gordon Moore died in March this year. And Robert Noyce and Gordon Moore wanted their own semiconductor company, and they reached out to Arthur Rock and Eugene Kleiner again. And Eugene Kleiner, the number 2 man, who at the time already got out of Fairchild and founded the legendary venture capital firm Kleiner Perkins to invest in their new company named Integrated Electronics, short Intel. The year was 1968. About 8 years later, Eugene Kleiner and Thomas Perkins got another investment opportunity of their lifetime. It was a company with nothing but a professor and a former Kleiner Perkins employee. The name of the professor with the prototype recombinant DNA technology was Herbert Boyer, the man on the right, and the VC hustler on the left was Bob Swanson. Kleiner and Perkins thought this was a huge bet with a big risk, but anyway gave them the seed money. In 1979, Genentech, led by Dr. Clyde, successfully applied the recombinant DNA technology and was able to produce massive amounts of insulin from bacteria and developed numerous miracle cures for various diseases. FYI, the number of pigs and cows killed in a year to meet the American insulin demand before Genentech was about 56 million. Spent too much time explaining the history. Now, the market size of chips and shots are similar. Semiconductor market is expected to reach about more than $700 billion in 2027, and biopharmaceutical market is expected to get closer to $700 billion around the same time. Both industries consume so much money for R&D and manufacturing costs. The worldwide semiconductor capital expenditures is going to reach about $170 billion. And pharmaceutical R&D expenditures are expected to get closer to $300 billion as we are proceeding into 2030. Even the business structure or the value chain steps are similar. I'm assuming that is why Samsung got into CDMO business in the first place. This is a lot of text, so let me walk you through. Sorry. We call the companies that create new chips as fabless, and the companies that create new drugs as bioventures, although startups usually sell their proven technology to big pharma. And the manufacturing process requires a rigorous procedures and cutting-edge equipment and materials. We call the companies that fabricate the actual physical chips as foundry, and the companies that manufacture the actual biopharmaceutical products under the guidance of FDA and EMA as CDMO. And there are firms that supply these producers, and standardized products in chips include memory chips such as DRAM and NAND flash, and in bio, chemical synthetic drugs and biosimilars. And next, Deng Xiaoping and Jimmy Carter in 1979. I think this picture signifies the beginning of US-China honeymoon period. It's been about 40 years. And on the right side, Donald J. Trump and Xi Jinping in 2018 at Beijing. Why these pictures now all of a sudden? It's because now China became so powerful in manufacturing over so many industries. So Korea now possesses edges over China only in semiconductors and biopharmaceuticals. And in our national interests, the US-China strategy competition is kind of an advantage for now. I ain't gonna lie, to be honest with you. The US restrictions are giving us more time to maintain our technological lead over Chinese firms backed by government subsidies. And it seems like it's going to be that way for some time. The Chips and Science Act of 2022 of the United States, the basic blueprint of that law was released in 2019 by the National Security Commission on Artificial Intelligence with funding from US Department of Defense with the purpose of winning the strategy competition with China. And this sentence, I think, shows how the brains and experts or strategy group in DC understand the current competition and expresses their resolution. This quote is actually a paraphrase from the quote by Winston Churchill in World War II. So I was talking about the fact that China has become a powerful nation with its prowess in manufacturing capabilities, and they need to catch up with Korea only in manufacturing in cutting-edge chips and shots. But it turns out not only China, but also in the Pacific nations, including India, Indonesia, and South American countries like Mexico, they all have great talent, population, technology, and they won't get their fair share in chips and shots. But let me assure you that making a foothold into these high technology sectors is very hard. And in our perspective, maintaining market share and coping with relentless competition and fast progress of technologies being developed are so much harder. So my main point is biopharmaceutical industry, just like semiconductors, is one of the most important and strategic field in Korean government's eyes, and we are serious about supporting not only Korean firms, but cooperating with corporations, people from allies and partner nations. Last year, with the President, Mr. Yoon, Korea passed the Korean version of Chips and Science Act of 2022, a big tax breaks and infrastructure packages for chips, shots, batteries, and the next generation display products. Our vision is to create a vibrant and dynamic innovation hub where miraculous new medicines could benefit not only the lucrative US and European market, but also Asian people with serious illnesses. In fact, Korea is famous for the miracle of Han River. That project was mainly confined to fostering big Korean businesses, not much caring about a truly multinational and not discriminative environment in our nation for people from various countries and backgrounds to freely work and enjoy their lives and create something the world never saw before but needed so long a time. So the new Bio Valley in Songdo, Incheon is going to be a new experience or experimental challenge for Korea and might be one of the most important steps to be a truly open and innovative nation. That vision of incubator now expands outside Incheon. In Seoul City and Korean Silicon Valley, Pangyo, there are numerous startups for drugs, chips, digital healthcare, finance, artificial intelligence. It only takes an hour or two to get there from here. At the end of the day, Korea is a small country. The density of talents per square meter be one of the best, I guess. Main message again, Korea will help and nurture biopharmaceutical industry as it have been doing for semiconductor for the last 40 years. Even the president and the cabinet, key players like Samsung, Lotte, Celltrion, and other big companies want to get in. The support packages you can check up on now, let me assure you, are only the beginning. The picture on the right side was taken when President Yoon visited SK Bioscience Research Lab. Final thoughts. Why investing here and hiring people is going to be a great choice. A little bit of deviation to make my point. Last summer, we had the 19th Asian Games in Hangzhou, China. And please appreciate the meaning of the distribution of medals each country got at the end of the game. So much medals for China. Korea and Japan, unlike China with 1.5 billion people, the talent pool is limited. But because Koreans are world best in private education spending, if the best talents are put into some sector or industry, after 10 or 20 years later, you see some amazing businesses pop up. Back in the 1960s to 1980s, Korean people got nothing but spirit and stamina. So we won gold medals in sports like marathon, wrestling, boxing. Now the young people does not want to fight. They got soft. So from 20 years ago, they got into swimming, fencing, shooting, golf, and so on. About 40 years ago, the best students in Korean high schools got into physics, electronics, chemicals department of universities. Now you see where we are in semiconductors. But since the 2000s, the absolute best students went into the medicine. It's partly because as Korea loses its edge in manufacturing sector and automation kicks in, the most well-paid jobs became doctors and pharmacists. Korean parents even now want to get their children to go to medical school. They spend literally $1 million to get them in. It's not a great phenomenon for national level, but turns out good for bio industry. Yeah. So in metaphorical sense, the potential gold medalists or champions in biopharmaceutical industry are now in hospitals and bio companies in South Korea. I'm sure they are the best, and you can feel it on day one if you hire them. The problem is maybe too smart to be controlled or micromanaged. In bio industry, I think costs are secondary. The talents are the most important thing. It's about technology, talent, and integrity, and most importantly, morality. Lastly, the reason why the next bio revolution is important is not entirely about making money. Innovation hub like Valley in San Francisco, the geniuses, the truly God-given talented people were not just about making profit. I'm not sure if you saw this movie, Dallas Buyers Club, starred by Matthew McConaughey. It's about him getting infected by HIV and trying to find his way to live, trafficking good working drugs from a doctor in Mexico who lost his US medical license selling the drugs. Obviously not approved by the FDA. And on the right, this kid named Incheon died waiting for the one-shot miracle cure for an acute leukemia named leukemia by Novartis, available in Korean market. It's kind of odd for me to preach people about not prioritizing money on a stand for a business conference, but let me tell you that Koreans are interested in making miracles, not just making money off of the unfortunate people. And that ends today's talk. Thank you very much for this.
Thank you for your presentation. Do you have any question? Feel free to ask. While you're preparing the question, I will ask a general question. The semiconductor business structure and bio business structure is somewhat different. Based on your semiconductor experience, what do you think would be first factor or required steps for bio business success or bioeconomy prosperity? Yeah, pretty general question, but somewhat you already explained about this. Yeah.
So let me tell you that the structure of economy of South Korea, Japan, and China is similar. They are all export-driven economy, and they have specialized in mass-producing products. So in semiconductors, Samsung got their success in mass-producing DRAMs, and NAND chips. And in biologics industry, they are trying to, it's my personal opinion, but they are trying to benchmark TSMC in Taiwan. So both our manufacturing side and Korea, Japan, and China are all weak in creating new products, new chips, new drugs. So, yeah.
Yeah, I also think the similar things like you, manufacturing, the mass production, one of the key factor. Also, I think, how about the investment? Investment, I think, investment for the semiconductor industry, the bio company, the bio-related business, I think we need to invest more. So.
Oh yeah, that's natural conclusion from the structure of the economy. Since you lack the ability to make innovative new products, and then you go into production, then you have no choice but facing price competition. That means you gotta make unit product at much lower price than China, than Southeast Asia countries. That needs massive investments.
Well, that's why we invited the investment expertise as the next speaker anyway. But do you have any other questions? Oh, maybe you explained very thoroughly and well. Thank you for your presentation again.
Yeah, I'd like to invite our third speaker. Okay. I'd like to introduce Dr. Su. 이게 아니지. Okay, sorry. Haesun Kim, who is the CEO of Taya SI Partners, and Ms. Haesun Kim serves as a CEO of Trans Globe Asia for 25 years, and consulting for various companies from small size to medium size. And she is also on advisory board for the many Korean governments. So today she will talk about how to position Korea's bio industry in Northeast Asia and attract foreign investment. Please give her big hands.
Thank you very much for your introduction. Sorry. Oh. So it's my great pleasure to present my presentation, and I very much appreciate Professor Kwon for inviting me to deliver my speech. So as I was introduced, I spent more than 25 years helping small and medium companies to grow in the international market, and also worked with many different Korean government organizations. So my presentation title is How to Position Korea's Bio Industry in Northeast Asia and Attract Foreign Investment. So this is my vision statement. I'm sure that all of you have some sort of vision, but because I like this, I have introduced vision without action is merely a dream. Action without vision just passes the time. So vision with action can change the world. So I'm sure all of you here are working hard to change the world. So where is Korea located? In the center of Northeast Asia, and located next door to the second largest economy, which is China, and the third largest economy, Japan. And we can reach out to 147 cities, which has more than 1 million population. But if we extend our flight to 6 or 7 hours, we can reach out to billions of population. So that's the target that we should make in terms of doing business. So let me explain about the Korean government policy. I think the past is very important because that's why we have present and we have future. So since 1998, which was just after the 1997 Korean economic crisis, like IMF bailout crisis, the Kim Dae-jung government has started to foster the IT industry and like startup. That was in line with maybe Silicon Valley booming of IT. And this IFEZ, which is Incheon Free Economic Zone, was established in 2003. So this year marked the 20-year anniversary of IFEZ. At the time, we only had 3 FEZs in the whole nation. But now we have almost 18 free economic zones nationwide. And in 2013, during the Lee Myung-bak government period, the Green Climate Fund organization was established in Incheon. And now we have more than 15 international organizations in Incheon, nationwide. And since then, we were focusing not only IT, but medical, bio, convergence, IT, AI, and IoT. And now, during the President Yu's era, which only started like 1.5 years ago, we are focusing on international market. I think, and especially in the R&D sector, the government is stimulating and encouraging the R&D to collaborate with international R&D research institute. So if we look at the bio industry, the production amount stood at 21 trillion won in 2021. I'm talking about the Korean bio industry, which was only 10 trillion won in 2016. So it's an annual increase of about 17.8% over the last 5 years. In particular, during the COVID-19 period, Korea's growth potential in biopharmaceuticals has already been proven by its excellent virus test equipment and world-class capabilities to manufacture vaccines and therapeutics. So since the introduction of FEZ in 2003, the accumulated amount of FDI in Korea was, now is 20 billion US dollar. This is accumulated amount, so there's a big room to grow, actually. But in case of IFEZ, the foreign investment amount accounted for about 70% among the all FEZ. So you can see how much Incheon has performed. So foreign investment business have also grown from 3 at the time of establishment of FEZ to current 206 in free economic zone. So bio cluster, Songdo is by no means, we can call it, is a rising star of bio cluster. Because of the bio production capacity of about 80 companies, including samsungbaio로직스 and Celltrion, it reached 800,000 liters. This is the figure is as of 2022, and is comparable with the capacity in San Francisco and Singapore. I don't know whether this figure is correct. Maybe Kayan can correct if it's wrong, because every source of material that I researched has slightly different figures, so please bear that in mind. And by 2030, Songdo Biocluster is targeting 700 tenant companies and 20,000 employees. So as per many speakers has previously explained, we are hosting a number of international, not only international, but Korean, but also conglomerate like SK Bioscience and Lotte, Samsung, and Celltrion, etc. And also international companies like Merck and Sartorius are continuously expanding their large-scale research and development manufacturing facilities. And Lotte is also planning to move into Songdo 11th zone. So as of 2023, Songdo has will be securing the world's largest biopharmaceutical production capacity by city standard with a capacity of 1.16 million liters. So let's look at the global biotech market size. It is estimated at 1.37 trillion US dollar in 2022, and it's a compound growth rate of 13.96% from 2023 to 2030. And this is the list of 25 top biotech companies by market cap. And there are an active M&A deals going on in this sector. So Seagen, which is ranked 16, is being acquired by Pfizer for 43 billion US dollar. And the deal announced on March 13th and expected to close in the late of this year or early next year. And Illumina is another case of active M&A. And bioagriculture is also a very important sector of bioindustry. Sustainable farming and crop insect resistance are examples of biotech in agriculture. And worldwide, biotech in agriculture is anticipated to grow by 25.3 billion US dollar between 2021 to 2025. The reason being this sector can produce more crop yield with fewer resources, aid the environment by reducing the chemicals and runoff from crop production, and create food without the mycotoxins or allergens. So who dominate the global market? Of course, it's North America, because it's accounting for 37.76% revenue share in 2022, followed by Asia Pacific, which is generating revenue share of 23.8% in 2022. So it's one of the fastest growing industry. So as Samsung Biologics VP also made a comment about this increasing aging population. I think it's worldwide a serious problem. But again, this can also stimulate the bioindustry. And especially in Korea, China, and Japan, three major Northeast Asian countries experiencing a fast aging society. And this year alone, people who are aged more than 65 is occupying 18% already in Korea. And the other problem that we're facing is shortage and low birth rate. So this can be a very serious problem in future for shortage of the workforce. And this data environment, the OMOPM CDM, which is like open information of clinical test. It will also, one of the sectors that can stimulate development of new drugs, because more than 70 countries are sharing their patient data worldwide. So let's talk about the globalization. During the pandemic era, people were talking more about domestic market, how to stimulate the consumption in the domestic market because of the lack of meetings, face-to-face meetings and travel barriers. But now, maybe since the APEC meeting between the US and China, we don't want decoupling, no competition, etc. I think the other companies will also follow the internationalization and globalization. And now we need to focus more on value creation and adaptation of the new market, unlike the past market and globalizing concept. So how is the global marketing trend changing? I think the COVID-19 period was a very typical experience that even following the COVID-19 period, people don't tend to meet frequently than before. So social media, cross-border commerce, and mobile platforms are becoming a strong tool for the marketing without having to meet face-to-face and lessen the number of meeting cases. So who are your competitors in the global market? And there are multiple shareholders in many different companies. For example, in many biomedical companies in Korea, the startup companies, especially in medical sector, there are many shareholders in one company, which make it sometimes difficult to make a decision in terms of many business activities. For example, there's a company that I was advising, and they had two different co-CEO. And when it comes to making decisions, one person agreed to this decision, but the other, he was against the final decision. So, the company management had difficulty in making decision in business activities. So why global? Where to go? What to sell? Korea has a 50 million population, but I think it's still a small country. So when you compare with China, with 1.5 billion, India or Indonesia, 270 million population, Korea is heavily depending on the export-driven economy. So we need the marketing and international expansion. And we need to find a local partner, target market, etc. So what will be the solution to globalize? My idea is to create an ecosystem. So market understanding is very important, and narrowing the gap between the target country and Korea is very important. But in many cases, many Korean companies, they are satisfied with Korean market, and they are satisfied with listing at the Korean stock market, which is very different from going out to international market and international listing. So, and the other important factor is raising fund, especially for small and medium companies. If they're expanding market, you need not only the partner, but need to raise fund at the right time, at the right timing. And it's very important to find a trustworthy partner. You cannot make a partner in one day. You need to build relationship, and it takes time to build relationship and trust. And the other factor is, of course, to get the certification for local market. One of the companies that we were advising was getting the FDA and CE. And when we were raising funds from international fund, the international investor was asking whether you have a FDA. The company has the FDA. And he said he's going through the FDA, but we have a KFDA, and they were asking for FDA. So the company owner said that it's more difficult to get the KFDA than FDA, because Korean FDA would add more regulation on top of FDA. So one of the options that was for Korean company was listing abroad. And in case of listing at NASDAQ, there are not many cases. Compared to other countries like China, there are more than 200 companies who are already listed at NASDAQ. And for example, Israel companies, the Israel population is under 10 million, but the companies who are listed at NASDAQ is over 200. So for Korean companies who are satisfied at Korean listing, why don't you think about listing at NASDAQ? But in order to list at the international market, you need to get a very top level business service people to help you. I just gave you some example of spec listing at the Singapore market. I know Singapore market is not a big market, but in order to activate and to raise fund, it can be one of the options for you. So if we look at the global R&D investment size, the Swiss giant Roche is spending $14.7 billion in R&D. And Merck and companies also allocated $13.5 billion. If we compare this amount with the Korean annual R&D spending, it's really a big difference. The Korean government is only spending $5 billion a year for R&D. But even that budget is shrinking, I'm afraid. So you can compare with the global big pharma companies, the size of the R&D investment. So what advice can I give to the Korean government? Especially in Incheon, I'm advising Incheon city government as a bio committee, global city committee. And they want to form an international fund, not only between the city government budget, but with private sector, private equity fund, together with the international matching fund. And I've been traveling around to find a way how to match this fund together. And the first question they're asking is, do you have a specific target company or target project? And where will you structure the fund? Who will manage the fund? There are a number of questions that you need to answer in detail in order to form a fund. And especially, there are a number of matching funds that is possible to match together with Korean fund, but those questions should be answered. And especially, I have been talking with one of my friends at the GIC in Singapore. He's in the infrastructure department, investment department, and he said that they are investing into Korea, but not alone. They are partnering with some international funds, such as KKR or Macquarie. And the investment size is $200 million US dollar. That is the minimum size of investment that they will make. So in order to attract funds for Korean small and medium companies, it's too much. We have to target different entities who are interested in working with the Korean government or Korean PE to invest into project. So my suggestion is that with the international companies, we should start with the R&D project together, and then attract local fund, or we invest into these projects. So there's no shortage of international network. We need to build international network, not only for marketing purpose, but also for investment and fundraising, etc. So let me just close my presentation with introducing a few companies that we are advising. I'm currently working as a partner of a venture capital fund based in Korea called K-Ground. And these are the portfolio companies that they have already invested. It ranges from semiconductor, battery, mobility, healthcare, etc. And in case of Nemesis, they are developing like semiconductor technology-based chips that will be embedded to mobility healthcare devices. And in case of Bonotox, they are like cosmetic company in Korea, but they have developed a special technology which is like a thin film type of facial mask that will, if you put on your face and spray with water, it will dissolve automatically. And NF is an oxygen supply system which can replace with existing oxygen tanks installed in many hospitals. And this is a special technology that extracts oxygen from the air. And it doesn't have to replace like ordinary oxygen tanks. And we have succeeded in exporting the product in the international market, like in Indonesia, CIS countries who are in need for this kind of medical product. So I have been to a Switch conference in Singapore early this month, and it was very interesting that the networking forum, so many international business people was gathering together. So I strongly encourage Incheon and Korea to network actively in the international market, and I hope to see many successful business people who can become the unicorn company in the international market. Thank you very much for your attention.
Thank you for the presentation, Ms. Kim. And any questions from the floor? Okay, then I'd like to add one question. You recommend several strategies for Korea companies and researchers in order to enter the global market or global investment environments. But what do you think is the weakest factor in terms of attractiveness for foreign investment?
That's a very quick question. There are many weakest points, especially for Korean companies. They are very concerned about their technology. And I think the most important factor for them is technology. But technology is only less than 20% of the whole business. You need to show them what is your target market, how will you raise, you will expand your market? What is your marketing strategy? What is your branding? And how will you position with your competitors in the international market? So for example, if there's one company in Korea, you need to understand, for example, if your company is listed in NASDAQ, for example, then you need to come up with like 10 different competitors. And you need to explain, you should be able to explain what is your competitive edge against those international companies. So it's and also the IR presentation material is very weak. It's not a matter of just translating into English, it's a matter of delivering your strategy.
Thank you. You mentioned that the technology itself is kind of strong in Korea, but
Yes.
we need to develop the package.
Package.
I see. Is there any question? All right, thank you. Please give a big hands to him, to her.
Thank you very much.
Okay, now is our last speaker. Okay, I'd like to introduce Dr. Soo-young Lee. He is head of New Drug Research Division of Celltron in Korea. Dr. Lee has served as head of New Drug Division since April 2021 in Celltron. Dr. Lee was promoted to Senior Vice President in 2021. And then he took the role of project leader to develop world first monoclonal antibody biosimilar, you already know, Lemsima, and the R&D project general manager for COVID-19 Therapeutics, which was marketed in Korea already, and also other countries including EU. Before entering to the Celltron, Dr. Lee was a senior researcher of Korea Green Cross Central Research Center and involved in developing many recombinant proteins. Okay, floor is yours, Dr. Lee.
마지막 스피커는 한국말로 진행을 하겠습니다. 저희 세 번째까지는 영어로 진행을 했는데 저기 통역분 앉아 계시는 게 심심하신 것 같아가지고 한국말로 진행하겠습니다.
Yeah, thank you for your introduction, and I think it's more comfortable to explain in Korean language. Most people think Korean people, so I will present in Korean today. 오늘 제가 발표드릴 내용은 우선은 지난 팬데믹 상황 이후로 바이오 헬스케어 인더스트리의 전반적인 상황에 대해서 말씀을 드리고, 그다음에 요새 언론에 자주 소개되고 있는 그 렘시마 SG, 미국에서 판매명은 진펜트라입니다. 진펜트라에 대한 소개를 좀 드릴까 하고요. 그리고 저희가 지금 저희 메인 비즈니스는 잘 아시겠지만 바이오시밀러 비즈니스입니다. 근데 지금 바이오시밀러 비즈니스에서 아까 계속 말씀을 하셨지만 그 신약 쪽으로 방향 전환을 하고 있고, 저희가 어떻게 신약 개발을 하려고 하는지에 대한 설명을 좀 드리도록 하겠습니다. 그리고 지금 현재 셀트리온이 부닥쳐 있는 상황에서의 그 위험 요소는 무엇이고, 그다음에 어떻게 저희가 성장하려고 하는지에 대한 설명을 드리도록 하겠습니다. 잘 아시겠지만 그 계속 글로벌 제약 시장의 크기에 대해서 그 설명을 할 때 자동차 시장만큼 크고, 그다음에 아까 세미컨덕터 발표하신 분 그 규모보다는 조금 작은 것 같은데 세미컨덕터 시장 2배 정도이기 때문에 상당히 큰 시장입니다. 그렇지만 저희가 그 글로벌의 제약 시장의 진출이 상당히 늦은 편이고, 잘 아시겠지만 전통적인 합성의약품 시장에서 원료 공급하는 그런 상황이었고 이제 근래 들어서 신약으로 2개 이상 지금 허가를 받고 하는 상황이기 때문에 글로벌 제약 시장의 메인 파인 어떻게 보면은 가장 큰 신약 시장에서의 시장에는 아직 발도 제대로 크게 들여놓지 못했다고 이해하시면 될 거예요. 이제 막 신약 시장에 진입해 가는 단계라고 이해하시면 될 것 같습니다. 그렇기 때문에 더욱더 앞으로가 기대되는 분야라고 저는 생각을 합니다. 잘 아시겠지만 글로벌 제약 시장의 한 70대 30으로 지금 합성의약품 시장과 바이오로직스가 구성되어 있고 이 바이오로직스의 규모가 한 580조 정도 되는데 그 중에서 작년 기준으로 셀트리온과 삼성바이오에피스 어떻게 보면 글로벌 시장에서 의미 있는 바이오로직스 시장을 형성하고 있는 그 분야일 것 같은데요. 거기에 전체 시장이 50조 원 규모를 형성을 하고 있고 2032년까지 160조까지 급속하게 성장할 걸로 예상을 하고 있습니다. 이 시장의 급속한 성장은 잘 아시겠지만 그 면역 항암제인 키투르다나 옵디보 같은 제품들이 2028년, 29년 그 무렵에 특허가 만료가 되기 때문에 그 시장을 고려했을 때 급속한 성장이 예상되고 있다고 그렇게 이해하시면 될 것 같습니다. 바이오의약품 시장은 초기에 잘 아시다시피 백신이나 인슐린, 그다음에 이포장 같은 1세대 바이오시밀러 제품들이 먼저 상업화가 됐고 이 제품 시장도 여전히 큰 포션을 차지하고 있습니다. 그렇지만 1세대 바이오의약품 같은 경우는 바이오시밀러 경쟁도 있고 진입 장벽이 비교적 이후에 말씀드리는 항체 바이오의약품 시장보다는 그 낮기 때문에 상당히 그 아까 해주셨던 대로 산업에 있어서 그 특성이 뭐 매스 프로덕션화되고 그다음에 코스트 경쟁력을 갖추지 못하면 퇴출되는 그런 상황으로 이제 점점 가고 있다고 생각을 하시면 될 것 같고 특히 백신 시장 같은 경우는 메이저 그 백신 회사들이 지배하고 있는 시장이었는데 이제 코로나를 겪은 뒤에서 설명을 드리겠지만 모더나나 바이오엔텍 같은 그리고 뭐 바이오엔텍하고 화이자가 같이 개발한 그런 코로나 백신 때문에 좀 판도가 지금 흔들리고 있다고 그렇게 이해하시면 될 것 같고요. 그 항체 의약품으로 대변되는 2세대 바이오 의약품 시장은 여전히 지금 성장하고 있습니다. 많은 제품들이 항체 그 바이오 의약품으로 신약 개발 단계에 있고 주로 항암 분야, 면역 분야에서 지금 시장을 갖다 계속 크게 성장시키고 있는 분야라고 이해하시면 될 것 같고요. 다만 이 시장 같은 경우는 잘 아시다시피 동물세포 배양에 기반한 그 매스 프로덕션 설비, 그다음에 플랫폼 기술을 갖추고 있는 회사들의 어떻게 보면 제한된 그 영역이라고 생각을 할 수가 있기 때문에 뭐 제네릭이나 1세대 바이오시밀러보다는 그 진입 장벽이 좀 높고 그렇기 때문에 그 정도의 경쟁까지는 안 되지만 지금 잘 아시다시피 뭐 삼성과 저희도 있지만 글로벌하게 보면 또 바이오시밀러 시장에 진출하고 있는 기업들이 다수 이제 생기고 있기 때문에 여기도 좀 경쟁이 심화되고 있다고 이해하시면 될 것 같습니다. 그리고 면역 항암제 같은 경우는 키투르다와 옵디보가 지금 연간 매출이 10억 불 이상으로 100억 불 이상으로 거대 시장을 형성을 하고 있고 여전히 계속 성장을 하고 있습니다. 그 이유는 키투르다 같은 경우는 인디케이션을 지금 계속 확장을 하고 있고 작용 유전상 다양한 암에서 효능을 발휘할 수 있는 포텐셜이 있기 때문에 계속 성장이 가능한 분야로 이해하시면 될 것 같고요. 세포 치료나 유전자 치료제는 최근에 이제 그 상업화가 이루어지는 분야인데 이 분야에서 가장 주목할 만한 것은 그 한 사람당 치료 비용이 크게는 5억 이상 되는 뭐 20억 되는 그런 제품들도 있고 한데 그만큼 어떻게 보면 고부가가치 산업 분야라고 그렇게 이해할 수 있을 것 같고요. 다만 이 분야에서는 그 자가 오토로거스하게 그 T 세포를 갖다가 콜렉션 한 다음에 그 환자한테 다시 투여를 하는 그런 불편함이 있고 생산 과정이 좀 복잡하다는 기술적인 어떻게 보면 진입 장벽이 좀 있을 것 같습니다. 요 테이블은 2022년 아까는 이제 그 스탁 마켓에서의 그런 그 가치로 지금 평가를 한 건데, 요거는 매출액 기준으로 보면 작년 그 2022년 기준에 봤을 때 사실은 2022년 말서부터 엔데믹으로 갔기 때문에 코로나가 이제 끝나간다고 그렇게 이해하시면 될 것 같은데, 여전히 작년까지는 화이자나 모더나 그다음에 바이오엔텍 같이 코로나 특수를 누리는 회사들이 여전히 강세를 보이고 있고요. 그렇지만 이 회사들이 엔데믹을 겪으면서 이제 회사가 그 세일즈가 줄어드는 거를 다른 걸로 이제 메꿔야 되는 상황이기 때문에 전략적으로 지금 M&A를 계속 진행을 하고 있고, 앞서 설명하셨지만 화이자 같은 경우는 다양한 그 분야에 자기의 파이프라인을 강화시킬 수 있는 M&A를 갖다 계속 지속적으로 하고 있고, 뭐 얼마 전에 시젠 같은 경우는 ADC 개발회사인데 시젠도 인수한다고 그렇게 기사화됐습니다. 요거는 이제 2022년 기준에 제품 탑10 세일즈 리스트를 보면 여전히 앞서 설명드린 대로 코미탄이나 스파이크박스, 그다음에 팍스로비드 같은 코로나 제품들이 10위권 안에 포진해 있고요. 그렇지만 올해 들어오면서 급격하게 지금 세일즈가 감소하고 있는 경향을 보이고 있습니다. 그리고 휴미라나 그 키투르다, 앞서 설명드린 스탈라라나 이러한 제품들이 사실 그 메인 저희가 바이오로직스 중에서 항체, 그다음에 제조합 단백질로 지금 큰 시장을 형성하고 있는 제품들로 이해하시면 될 것 같고요. 키투르다 같은 경우는 단일 품목으로 210억 불 정도 지금 세일즈를 내고 있고, 앞서 설명드렸듯이 인디케이션을 계속 확장하면서 2030년까지 400억 불까지 늘어날 걸로 그래 예상하는 마켓 그 레포트도 있습니다. 이 중에서 이제 휴미라, 그다음에 스탈라라 이러한 제품들은 저희가 지금 바이오시밀러로 개발을 하고 있는데, 휴미라 같은 경우는 올해 이제 특허가 미국에서도 만료가 됩니다. 그래서 세일즈에서 아직까지는 그렇게 급격한 감소를 보이고 있지는 않지만 앞으로 향후에 점점 더 시장이 이루어져되면서 휴미라의 세일즈는 줄어들고 그 바이오시밀러 제품들의 그 세일즈가 급격하게 성장할 것으로 이렇게 예상을 하고 있습니다. 질환별로 보면은 뭐 잘 아시겠지만 코비드 19이 엔데믹으로 가고 있지만 여전히 세일즈에서 앞서 설명드린 대로 강세를 보이고 있고, 온콜로지가 그 앞서 설명드렸던 키투르다와 같은 면역 항암제의 급속한 세일즈 성장으로 인해서 가장 세일즈가 높은 분야로 적응증을 보이고 적응증이고, 그다음에 안티바이오틱, 그다음에 자가면역 질환인 류마티즈 관절염 같은 부분이 2, 3위를 달리고 있는데, 특히 앞서 설명드렸듯이 위고비나 마운자로 같은 그런 원래는 당뇨를 타겟팅해서 개발이 됐지만 비만 효과를 보이는, 비만에 대한 효과를 보이는 걸로 해서 지금 굉장히 세일즈를 갖다가 크게 확장시켜 나가고 있고, 그렇기 때문에 이제 당뇨 영역도 항암 영역만큼 성장이 가파르게 되는 적응증 분야로 이해하시면 될 것 같습니다. 램시마에 대해서 좀 설명을 드리고 뒤따라서 램시마 SC에 대해서 좀 설명을 드리도록 하겠습니다. 램시마는 2013년에 그 EMA에서 허가를 받아서 판매가 시작이 됐고, 2015년서부터 20년까지 그 미국과 EU 주요 5개국에서 그 바이오시밀러 론치로 인해서 의료비 절감 효과를 갖다가 분석한 결과 보고서에 보면 1,070억 달러 정도의 5년간 절감액을 갖다가 가져오는 큰 효과를 의료 산업에서는 큰 어떻게 보면 혁명적인 일이라고 생각을 하고 있습니다. 그리고 국내 바이오시밀러 시장을 갖다가 이제 결국에는 뭐 수출해서 다 버는 거니까요. 그 국내 바이오 산업도 크게 성장을 시켰고, 그 다음에 2017년 기준으로 해가지고 전체 바이오 의약품 시장 중에 한 45%를 형성하는 큰 영향을 갖다가 끼쳤다고 그렇게 이해하시면 될 것 같고요. 램시마 SC 진펜트라는 이러한 램시마 IV 제형을 갖다가 고농도 제형으로 개발한 제품입니다. 그래서 이 램시마 같은 경우는 인플렉시맵 TNF-알파 블라커인데 이 전체 시장은 500억 불 정도 되는 시장입니다. 그 중에서 이제 저희가 기존의 레미케이드가 갖고 있지 않은 램시마 SC 제형을 갖게 됨으로써 이 전체 TNF-알파 블라커 시장에서 새로운 옵션을 제공할 것으로 이렇게 기대하고 있습니다. 특히 저희가 먼저 미국 시장에 진출하는 건 IBD 질환 쪽인데 IBD 질환에서는 처음에 IV 인젝션 후에 SC로 메인티넌스 할 수 있는 그러한 의료적인 치료법을 갖다가 새롭게 제공을 함으로써 환자들에게 편의성을 갖다가 제공을 하면서 시장에서 포지셔닝 할 수 있을 것으로 생각을 하고 있습니다. 이러한 램시마 SC의 제형 특허는 2037년까지 독점적 권리를 보호받고 있고요. 그 다음에 어드미니스트레이션 메소드에 대한 특허는 2040년까지 독점적 지위를 갖고 시장에서 플레이할 수 있습니다. 이 그림은 EU 5개국에서의 마켓 쉐어를 갖다가 지금 보여주는 커브인데 그림에서 보듯이 저희가 원래 가지고 있던 램시마 IV와 램시마 SC 새로운 출시로 전체 인플리빅시 시장의 거의 70% 가까이를 지금 마켓 점유를 하고 있고요. 램시마 SC 제형 출시 이후에 그 성장 속도를 보면 지금 현재 전체는 18%인데 독일과 프랑스에서 각각 32%, 21%로 빠른 속도로 지금 시장을 잠식해 나가온다고 이해하시면 될 것 같고요. 다른 오른쪽 그림은 다른 같은 시기에 나온 신약이랑 비교했을 때도 빠르게 시장에 침투하는 것을 알 수 있습니다. 이러한 램시마 SC는 바이오베터의 일종이라고 생각하시면 될 것 같습니다. 여기에 그 바이오베터에 대한 어떻게 만들까에 대한 전략적인 컨셉을 갖다 여기서 설명을 하고 있는데 그 중에서 저희가 고농도 제형으로 해서 SC로 지금 편의성을 갖다 제공하면서 바이오베터 제품을 개발해서 판매를 하고 있다고 그렇게 이해하시면 될 것 같고요. 그 외에도 효능이나 안전성을 극대화시킬 수 있는 바이오베터 제품을 개발을 하거나 아니면 헬프라이프를 늘려주면서 환자에게 편의성을 제공해 줄 수 있는 바이오베터 제품을 개발을 할 수가 있습니다. 바이오베터 제품의 장점이라고 하면 이미 임상적으로 효능을 보일 수 있는 작용 기전에 대한 검증이 끝난 개발 리스크가 작은 제품이라고 그렇게 이해하시면 될 것 같고 그렇기 때문에 이제 오리지널 제품의 개발 과정에서 먼저 임상적인 디자인이나 이런 것들을 고려해서 저희가 임상 디자인을 하면서 개발 리스크, 그다음에 개발 기간, 그다음에 개발 비용을 갖다가 줄일 수 있는 장점이 있습니다. 그렇지만 바이오베터 제품도 결국엔 신약 패스웨이 임상 1상, 2상, 3상을 다 해야 되기 때문에 리스크 면에서의 장점은 있지만 어쨌든 규제적으로 큰 장점은 없습니다. 지금 바이오베터 제품을 갖다 저희가 집중해서 뒤에 좀 저희 회사의 신약 전략을 갖다 설명을 드리겠지만 개발하고 있는 건 안티바디 드럭 컨주게이트 그다음에 바이 스페시픽, 멀티 스페시픽 안티바디 두 분야라고 그렇게 이해하시면 될 것 같고요. 이 안티바디 드럭 컨주게이트는 잘 아시다시피 톡식한 페이로드를 안티바디를 통해서 타겟하는 암 세포 표면에 항원에 전달함으로써 세이프티 면에서 기존의 톡식한 페이로드의 세이프티를 세이프티 이슈를 갖다가 최소화시키면서 항체만 사용했을 때는 효능적으로 좀 미흡한 부분을 이러한 톡식한 페이로드를 쓰면서 효능을 극대화시킬 수 있는 그런 제품으로 그렇게 이해하시면 될 것 같고 앞서 설명드렸던 시젠사와 같은 경우는 지금 혈액암에서 주로 ADC 제품을 상업화해서 성공을 했다고 그렇게 이해하시면 될 것 같고, 작년인가요? 작년에 아스코에서 그 굉장히 의미 있는 임상적 결과를 유방암에서 보여줬던 에너투라는 제품은 아스트라제네카가 지금 다이치 산교랑 같이 공동 개발을 해서 상업화를 성공했습니다. 그리고 바이스페시픽 안티바디 같은 경우는 듀얼 타겟팅을 하거나 아니면 T셀 인게이저를 통해서 효과를 극대화시키는 그런 항암제 또는 여기에서 보듯이 그 습성 그 황반 변성증에 대한 그런 제품들이 지금 상업화돼서 팔리고 있습니다. 그 특히 지금 J&J에서 개발한 택발리 같은 경우는 지금 혈액암 멀티플 마이엘로마에서 굉장히 의미 있는 임상 결과를 보여주고 있고, 존슨앤존슨 외에도 지금 그래서 이중항체 굉장히 집중을 많이 혈액암 쪽에서는 하고 있는 모달리티입니다. 그 잘 아시겠지만 아까 카티 설명을 하셨는데 카티에서 보이는 그 효능은 상당히 의미가 높은 그런 효능을 보이고 있거든요. 오버롤 리스판스 레이시 막 90% 이를 정도로 효능 측면에서 굉장히 뛰어난 효능을 보이는 그런 그 임상 결과들도 나오고 있는데, 이러한 이중항체도 그거에 못지않은 효능을 보이고 있고, 특히 대부분의 카티 치료제는 이제 오토로그스로 아까 말씀드렸듯이 환자에서 T세포를 갖다가 콜렉션 한 다음에 다시 이제 넣어주는 형태이기 때문에 굉장히 제조 과정이 까다롭고 복잡하기 때문에 이 이중항체에 비해서는 그런 불편함이 있습니다. 그래서 이중항체가 혈액암에서 새로운 포지셔닝이 가능한 그런 모달리티로 그렇게 저희는 이해하고 있습니다. 그 외에도 글라이코 엔지니어링을 통해서 반감기를 늘려주는 그러한 바이오베터 제품도 있습니다. N2에 대한 거는 앞서 설명드렸지만 여기서 보듯이 보통은 허투가 많이 발현된 유방암에서 저희가 타겟팅을 했는데, 이 N2의 가장 큰 의미 있는 결과는 캐살라 대비해 가지고 워낙 높은 오버롤 리스판스 레이시 보인 것도 의미가 있지만 허투 로우에 대해서 효능을 보였다는 것은 굉장히 의미 있는 일이고, 이거는 여기에서 토포 아이소메라즈 이니비터로 사용된 데룩스테칸이라는 이러한 페일로드가 그 바이스탠더 이펙트를 보이면서 이러한 결과를 보였을 걸로 예상이 되기 때문에 이 N2 결과를 봤을 때 고형암에서는 페이로드가 중요한 역할을 하지 않나 하는 생각을 하고 있습니다. 그래서 저희 같은 경우는 고형암에서는 좀 페이로드를 쓸 때 좀 레스 톡식한 페이로드를 써서 DAR을 드러그 안티바디 레이셔를 극대화하게 되면 좀 더 고형암에서 효과적으로 반응할 수 있을 거라고 생각하고, 국내 피노바이어라는 기업과 지금 공동 연구를 진행을 하고 있고요. 다만 혈액암에서는 앞서 설명드렸던 시젠사도 그렇고 대부분 그 MMA 같은 그런 약간은 이러한 데룩스테칸 페이로드보다는 좀 더 톡식한 페이로드를 쓰거나 아니면 PBM 같이 아예 굉장히 독성이 높은 페이로드를 써서 제품을 개발을 하고 있기 때문에 저희도 그러한 컨셉으로 지금 혈액암에 대해서 개발을 진행하고 있는 익스다에 지분 투자를 하면서 향후에 이러한 부분에서도 전략적으로 저희 혈액암에서는 이러한 어프로치로 지금 개발을 진행을 하려고 하고 있습니다. 저희의 가장 큰 골은 일단 플랫폼을 확보하고 그다음에 파이프라인을 확보하기 위해서 코디벨롭을 하거나 라이센스 인을 하는 것입니다. 그리고 그 바이스페시픽, 멀티스페시픽 같은 경우는 앞서 설명드렸듯이 혈액암에서 T셀 인게이저를 통해서 지금 뚜렷한 효과를 갖다가 지금 보이고 있기 때문에 저희도 여기에 주목해가지고 이러한 플랫폼을 갖고 있는 회사와 공동 연구를 통해서 지금 혈액암 쪽에서 그 파이프라인 개발을 갖다가 진행을 하고 있습니다. 저희는 잘 아시다시피 지금 현재는 바이오시밀러가 메인 비즈니스인 회사입니다. 그렇기 때문에 바이오시밀러의 현재 상황에서 저희가 어떤 전략을 가져갈지가 중요합니다. 지금 경쟁이 심화되고 있기 때문에 가격에서 경쟁력을 갖다가 확보해야만 글로벌 시장에서 저희가 넘버원 위치를 계속 유지할 수 있다고 생각을 하고 있고요. 그렇기 때문에 그러한 목적으로 지금 기사화되고 많이 이제 관심이 있는 셀트리온과 셀트리온 헬스케어 간의 합병을 통해서 저희가 개발, 그다음에 생산, 그다음에 판매까지 아우르면서 전체적으로 공급망을 수직 계열화시키면서 만들게 된다고 하면 저희가 가격을 플렉서블하게 조정할 수 있는 경쟁력이 생깁니다. 그런 가격 경쟁력에서의 플렉서빌리티는 결국에는 추후에 벌어지는 가격 경쟁에서 저희가 좀 더 낮은 가격으로 파일을 늘려갈 수 있는 그런 원동력이 될 거라고 생각을 하고 있습니다. 그리고 저희가 뒤에 설명드리겠지만 2030년까지 22개까지 파이프라인, 지금 현재는 6개인데 파이프라인을 지금 늘려갈 거기 때문에 아까 삼성바이오로직스에도 설명을 하셨지만 다품종 소량 생산이 필요하고 그다음에 하이 타이터를 통해서 저희가 공정 개선을 통한 하이 타이터를 확보를 하게 되면 지금 현재는 15,000리터, 12,500리터 정도의 그런 사이즈인데 미들 사이즈의 바이오렉터를 갖다 여러 개 두면서 거기에서 저희가 원하는 프로덕트를 갖다가 상업적으로 생산할 수 있기 때문에 그러한 전략으로 지금 제상 공장은 설계돼서 운영을 할 예정입니다. 그리고 지금 아까 설명드렸듯이 신약은 미래를 위해서 지금 준비를 하고 있고 지금 개발을 시작을 한다고 하더라도 잘 아시겠지만 중간에 라이센스 아웃을 통해서 그런 마일스톤 비용을 갖다 확보를 하지 않는 이상은 저희 세일즈에 직접적인 영향을 크게 당분간은 주지는 못하겠죠. 그렇지만 미래는 점점 더 컴피티션이 많이 심해지면 결국에는 저희가 신약을 통해서 저희가 가지고 있는 이 가치를 갖다 유지하기 위한 그런 제품들을 또 만들어내야 되기 때문에 항체 기반의 ADC나 그다음에 항체 기반의 멀티 스페시픽 항체를 갖다가 지금 항체 제품을 갖다가 파이프라인으로 계속 2024년서부터 지금도 계속 파이프라인을 갖다가 구축을 하고 있는데 빠른 시기 내에 임상을 개시할 수 있도록 파이프라인을 구축을 하고 있습니다. 그리고 기존 제품 같은 경우는 저희가 고농도 제형으로 해가지고 SC뿐만 아니라 자가 면역 질환에서 오랄 딜리버리가 가능한 플랫폼을 가지고 있는 회사라 공동 연구를 통해서 오랄 딜리버리 제품도 개발을 하고 있습니다. 그리고 비알코올성 간 질환에 대한 또 제품도 저희가 개발을 하고 있고요. 사실 이러한 부분 중에서 저희가 가지고 있는 향후에 에셋이 될 수 있다고 생각하는 게 지금 저희는 다수의 바이오시밀러 프로젝트를 진행을 하면서 글로벌 임상을 많이 진행을 하고 있습니다. 그래서 환자 동의하에서 환자 샘플을 활용을 해서 환자 샘플을 분석을 통해서 유전체 정보나 이러한 데이터베이스화 할 수 있는 바이오 인포메틱스 정보를 구축을 하려고 하고 있습니다. 그래서 이러한 데이터베이스가 구축이 되면 이러한 데이터베이스를 활용을 해서 AI 기술을 적용한 모델링을 통해서 의미 있는 바이오마커를 도출해 내거나 새로운 신약 개발을 그걸 통해서 새로운 신약 개발할 수 있는 후보 물질을 스크리닝하는 그런 새로운 미래에 대한 준비도 지금 진행을 하고 있고 결국에는 이러한 디지털 헬스케어의 목표는 프리시전 메디슨을 할 수 있도록 준비하는 것입니다. 그래서 여기에서 보듯이 앞서 설명드린 현재는 600억 불 정도의 6개 제품을 가지고 있고 2030년까지는 1820억 불 정도의 22개 제품 파이프라인을 라인업으로 갖게 될 계획입니다. 지금 순조롭게 지금 임상 허가 진행되고 있기 때문에 전체 계획에는 큰 차질은 없다고 이해하시면 될 것 같고요. 지금 셀트리온 헬스케어는 미국 셀트리온 USA가 지금 설립이 됐고 내년서부터는 직판을 할 예정입니다. 그리고 이외에도 다이렉트 세일즈가 가능한 셀트리온 헬스케어 지사가 있고 아시아 퍼시픽이나 다른 지역에도 지금 인다이렉트나 다이렉트 세일즈 네트워크가 구축이 돼 있어서 현재 110개국에 지금 저희의 판매망이 있다고 그렇게 이해하시면 될 것 같고, 이러한 판매망은 바이오시밀러 제품도 있지만 향후에 신약 개발이 이루어졌을 때 엄청난 파워를 갖게 될 걸로 그렇게 예상을 하고 있습니다. 앞서 설명드린 대로 저희가 준비하고 있는 제상 공장은 7,500리터 8기로 하이 덴시티, 그러니까 하이 프로덕티비티, 그러니까 생산성을 높여가면서 이러한 미들 사이즈 바이오리액터에서 상업 생산이 가능하도록, 그리고 다품종을 커버할 수 있도록 준비를 하고 있다고 이해하시면 될 것 같고, DP에 대한 설비도 지금 자체적으로 익스펜션을 하고 있습니다. 그리고 글로벌 네트워크도 EU나 아시아, 미국 지역에 지금 파트너십을 통해서 갖추고 있기 때문에 전체적으로 바이오시밀러 생산 캐파에 대한 충분한 준비가 되어 있다고 그렇게 이해하시면 될 것 같습니다. 새로운 신약에 대한 준비는 주로 자가 면역 질환, 그다음에 캔서, 그리고 이제 대사 질환에 대해서 지금 공동 연구 개발을 통해서 파이프라인을 확장해 나가고 있다고 이렇게 이해하시면 될 것 같고, ADC에서는 앞서 설명드린 피노바이오나 익수다랑 같이 하고 있고, 이중항체 같은 경우는 국내 기업 진유부나 아니면 사이런 같은 회사랑 지금 하고 있고, 국외 기업 같은 경우는 에비프로사랑 지금 공동 개발을 하고 있습니다. 그리고 라니사랑 해서 오랄 딜리버리 오토이민 디지즈에 대한 제품 개발을 진행을 하고 있습니다. 셀트리온 헬스케어 인텔리전스 뱅크라고 저희가 명명을 했는데, 이 셀트리온 헬스케어 인텔리전스의 가장 키는 저희가 가지고 있는 임상을 진행하면서 각 질환별로 저희 환자의 동의하에 샘플 분석을 통해서 얻어지는 데이터베이스라고 그렇게 이해하시면 될 것 같고요. 뭐 퍼블릭의 UK뱅크나 이러한 거대한 그런 데이터베이스들이 있기는 하지만, 그러한 데이터베이스들은 시결적이지도 않고, 그다음에 환자에 대한 질환 정보나 그다음에 투약 정보가 그렇게 상세히 정리가 되어 있지 않기 때문에 저희가 만들 수 있는 자체적인 데이터뱅크의 퀄리티를 굉장히 우수하다고 생각을 하고 있고, 이러한 데이터뱅크를 구축을 하고 활용하기 위해서 지금 현재 바센바이오랑 같이 공동 연구를 진행을 하고 있습니다. 그리고 이렇게 모아진 데이터뱅크를 통해서 AI 기술을 활용한 모델링 뱅크를 만들 계획이고, 이러한 모델링 뱅크를 통해서 결국에는 바이오마커 발굴이나 새로운 신약 후보 발굴에 활용할 예정입니다. 이렇게 된다고 하면 결국에는 저희가 궁극적으로 목표로 하고 있는 신약 그리고 바이오 의약품 분야에서의 프리시전 메디슨을 가능하게 할 걸로 그렇게 예상을 하고 있습니다. 네, 감사합니다.
네, 이수영 박사님 감사합니다. 그러면 플로어 질문 받겠습니다.
As we made this pitch in Korea, I also question in Korea. 네, 안녕하세요. 저는 백신 실용화학 기술개발 사업단에서 근무하는 박성우라고 합니다. 오늘 이 부사장님의 그 셀트리온에 대한 여러 가지 비즈니스 모델이나 이런 거 감명이 잘 들었습니다.
부사장.
예. 점점 더 바이오시밀러 마켓 쉐어가 늘어나게 된다는 것은 굉장히 고무적인 부분이고, 특히 기술적인 부분에 있어서도 지금 셀트리온이 현재 6개 파이프라인을 향후에 22개까지 이제 확대를 하겠다는 것은 철저하게 R&D 베이스가 뒷받침되는 그런 전략의 일환에서 나오는 거라고 저는 생각을 하고 있는데요. 코로나19를 통해서 경험했다시피 지금 mRNA 플랫폼이 사실은 시작은 백신에서부터 시작이 됐지만, 지금은 특히 이제 인베스트먼트나 글로벌 R&D에 있어서도 이게 굉장히 중요한 바이오텍의 플랫폼, 핵심 플랫폼 기술로도 굉장히 많이 지금 빠르게 지금 움직여지고 있고, 특히 스타트업이나 벤처들이 이 부분을 집중 공략하고 있는데, 혹시 이런 분야에 있어서 mRNA 플랫폼을 같이 콜래보레이션해서 R&D를 하거나 아니면 셀트리온 자체가 mRNA 플랫폼을 직접 R&D 할 수 있는 이런 거는 어떤 계획을 갖고 계신지 궁금해서 좀 질문드렸습니다.
mRNA 플랫폼에 대한 뭐 이거는 좀 개인적인 오피니언일 수 있지만 그 생각은 포텐셜이 상당히 크다는 거는 맞는 거이고 앞서 설명드렸듯이 뭐 지난 코로나 3년간 급속하게 성장을 했기 때문에 모더나, 화이자, 바이오엔텍이 포텐셜이 상당히 크다는 거는 저희도 이해하고 있고 그래서 사실 자체적으로 그 플랫폼을 갖다가 스터디하고 그다음에 기술 이전을 통해 가지고 자체적인 비임상 시료 생산까지에 대한 그런 기술 축적은 이제 해놓고 있습니다. 그래서 준비는 하고 있는데 가장 중요한 거는 사실 백신 분야에서의 그 저희랑 미스매치되는 부분 분명히 있고요. 그래서 저희가 지금 생각하는 거는 잘 아시겠지만 이제 mRNA 가지고 항체를 뭐 제약 쪽으로 해가지고 개발하는 업체들도 있긴 하지만 가장 이제 현실적으로 지금 모더나에서 지금 키투르다랑 병용해가지고 그 맞춤형 의학으로 항암에서 효과를 보였기 때문에 이제 그 가능성을 보고 지금 계속 임상 3상을 하면서 지켜보고 있는 중이라고 이해하고 있거든요. 그래서 일부 또 질환 뭐 희귀 질환에서 이제 mRNA 가지고 임상하는 것도 있고요. 근데 아직까지는 치료제 분야에서의 뚜렷한 mRNA의 그 가능성 확인은 되지 않았다고 저희는 이해하고 있고요. 그래서 지금 국내 기업 그 치료제 개발하고 있는 기업들과 미팅을 계속 하고 있기는 합니다. 그리고 근데 이제 그거는 저희가 검증이 어느 정도 되고 나서 저희가 기술을 가지고 있는 사실 아시다시피 그 mRNA 플랫폼 중에 그 LNP 기술이나 그 다음에 그 DNA를 갖다가 설계하는 기술들 이런 그 플랫폼들을 가지고 있는 기술 그 국내 회사가 그렇게 많지는 않거든요. 그래서 그런 회사들의 기술을 보고 있고 그 다음에 이제 트렌드로는 그 앞서 설명드렸듯이 이제 모더나에서 하고 있는 그런 그 항암 분야에서의 그런 것들이라든지 아니면 이제 희귀 질환에서의 그 가능성을 보고 있는 중이라고 이해하시면 될 것 같고요. 그러한 기술들이 검증이 된다고 하면 저희도 당연히 그 분야에서 좋은 기술을 갖고 있는 국내 기업과 공동 연구를 통해 가지고 지금 저희가 생각하는 건 키투르다나 옵티보 같은 그런 면역 항암제가 바이오시밀러 그 자체로 시장에서 당연히 의미 있는 그런 세일즈를 내는 것도 중요하겠지만 잘 아시겠지만 키투르다 같은 경우는 상당히 많은 병용 요법으로 지금 그 다양한 암 질환에서 효능을 계속 보여주고 있기 때문에 이제 mRNA 기술도 그런 식으로 이제 병용할 수 있는 파트너로 그렇게 활용 가능한 기술로 현재는 이해하고 있고요. 그 신약 기술도 지금 사실은 그런 키투르다 바이오시밀러 론치 이후에 저희 신약과 병용해가지고 특정 질환에서는 좀 더 뛰어난 효능을 갖다 보일 수 있도록 지금 결국에는 제품화가 돼야지 저희 신약도 의미 있는 그런 세일즈를 갖다 낼 수 있을 거라고 그렇게 이해는 하고 있습니다.
네, 또 다른 질문 있으십니까? 없으시면 제가 한 가지 간단하게 질문 드리고 싶은데요. 일단 바이오시밀러 일단 셀트리온이 제가 알고 있기로 그 글로벌 탑 50 바이오파마슈티컬 컴퍼니에 들어간 한국의 유일한 기업이에요. 매출 기준으로. 그러니까 삼성바이오로직스는 바이오파마슈티컬 컴퍼니로 구분이 안 돼서 하여튼 축하드립니다. 저도 굉장히 기뻤습니다. 그 통계를 보고. 근데 그 바이오시밀러 비즈니스가 경쟁이 심화될 거다라고 보신 거에 저도 동감을 하는데 그럴 때 가장 큰 경쟁 국가가 중국, 인도를 포함하는 건지 일단 그 질문하고 그다음에 이게 가격 경쟁력 같은 거를 확보하기 위해서 그 뭔가 직판을 하시기 시작했는데 그게 사실 이게 유통망이 그 의약품은 유통망을 갖기가 굉장히 보수적인 시장이라 어려울 것 같은데 그 부분에 어려움이 없었는지 두 번째 질문, 세 번째 질문은
두 번째 질문이 뭐...
첫 번째 질문은
네, 그건 아니고
예, 두 번째 그러니까 직판하려고 지금
네.
미국 시장이랑 유럽 시장에 그 여러 뭐 헬스케어 통해서 그런 유통망을 만들고 계신 거잖아요. 근데 이제 그 보수적인 글로벌 시장의 유통망을 과연 경쟁력이 있을 것인가? 왜냐면 뭐 존슨앤존슨이나 파이저나 이런 글로벌 그 제약사를 통해서 유통하는 것보다 직접 할 수 있는 것에 대한 어떤 경쟁력
네.
그 얘기를 잠깐 듣고 싶고 또 하나는 앞서 이제 정책 관점에서
네.
삼성바이오로직스는 인력만 있으면 된다.
네.
정부에서 지원할 건 탤런트의 문제다라고 하셨는데 혹시 셀트리온 입장에서는 정책 니즈가 뭐가 있나 말씀해 주세요.
네. 첫 번째 질문은 그 경쟁 그 상황에서 경쟁자로 지금 인도 기업이나 그 중국 기업이 포함되느냐의 질문은 포함될 수도 있고 안 될 수도 있다고 생각합니다. 지금은 바이오콘 같은 경우는 뭐 이미 그 비아트리스에서 그 바이오 시밀러 파이프라인에 대한 판권을 가지고 왔기 때문에 저희랑 경쟁을 할 수는 있겠지만 그 기본적으로 저희가 생각하는 바이오시밀러의 가장 큰 그 부분이 다른 것도 그렇지만 결국엔 일정 정도의 세일즈를 확보를 이미 하고 있는 기업이 가지고 있는 베너핏이 굉장히 많다고 저는 생각을 하고 있습니다. 그래서 제조 원가에 사실은 그러니까 인베스트먼트를 아까도 말씀드렸듯이 크게 하게 되면은 그만큼 이제 그 세일즈를 기대를 하고 하는 건데 그 세일즈가 형성이 안 되면 사실 그 인베스트먼트를 유지를 할 수가 없지 않습니까? 예를 들어서 10,000리터짜리 바이오리액터를 갖다가 투자를 해가지고 거기서 나오는 걸 팔아서 그 어떻게 보면 생산 코스트를 이제 다운 시키려고 뭐 타이터 높이고 이렇게 하지만 실질적으로는 판매가 안 된다고 하면 그건 다 허상인 것 같고요. 그래서 지금 그 인도와 인도와 중국 기업의 가장 큰 장점은 사실 그거거든요. 그 내부 안에서 도메스틱에서 판매할 수 있는 그 이게 굉장히 큽니다. 그러니까 거기서 이미 워낙 인건비도 싸지만 거기서 이미 그 생산 코스트를 줄일 수 있는 그런 판매들이 다 이루어지고 이제 외부에 보낼 때는 굉장히 싸게 보낼 수 있는 거죠. 근데 바이오시밀러도 마찬가지일 거라고 생각을 하고 있고 단지 이제 좀 바이오시밀러 같은 경우는 워낙 고가의 제품이다 보니까 아직 시장이 그렇게 성숙돼 있지는 않는 걸로 그렇게 생각은 하고 있습니다. 그래서 조금 그게 지체될 뿐이지 결국에 그 규모의 경제가 거기서 내부에서 이루어진다고 하면 그 다음에는 이제 규제 문제가 그 인도와 유럽에서 뛰어넘어야 될 건데 제가 보기에는 뭐 인도는 이미 사실은 바이오콘 같은 기업은 뛰어넘고 있고 거기는 사실 워닝 레터를 계속 받는 것 같아요. 주기적으로 워닝 레터 받는다는 얘기가 나오지만 미국에 판매를 못하는 건 아니거든요. 그렇다고 해서 그래서 결국에는 그 경쟁 구도에 인도와 중국이 들어오긴 할 텐데 지금 현재는 그런 규모의 경제를 만들어내기에는 바이오시밀러 제품들이 그렇게 그 안에서 활성화돼 있냐? 그렇게 보이지는 않은. 왜냐하면 저희가 느끼고 있으니까 인도에 바이오시밀러를 팔러 들어가면 너무 싸고 해서 과연 그게 사실은 문제가 될 것 같아요. 그 자체가 저희가 보기에는 오히려 이제 메인 시장에서 이게 엄청나게 판매를 하고 있는 저희가 그런 면에서는 오히려 생산 단가를 낮추는 데 유리한 조건을 갖고 있다고 이렇게 생각은 하고 있고요. 두 번째 질문이
정책적인 니즈 전에 그 유럽 시장이랑 미국 시장에서 직접 파는 거에 글로벌 유통망의 보수성을 깨기가 쉬우셨나?
그 부분에 대해서는 사실은 뭐 제가 그 셀트리온 헬스케어 쪽이 아니라서 정확하게 답변을 드릴 수는 없지만 유럽에서는 이미 이제 저희가 그 램시마라는 그 제품이 처음으로 출시되면서 바이오시밀러는 셀트리온이라는 브랜드화가 돼서 어떤 면에서 보면은 사실 그 바이오시밀러 영역에서는 그 존슨앤존슨이나 이런 데랑 완전히 다른 섹터니까 그런 걱정은 하지는 않고 있습니다. 그래서 사실은 텐더 시장이기 때문에 유럽 시장에서는 가격 경쟁을 갖고 있다고 하면 누구나 이제 그 위치를 갈 수 있지만 셀트리온은 그거에 플러스 알파로 이제 바이오시밀러에 대한 브랜딩이 이미 이루어졌기 때문에 환자 그다음에 의사 선생님들이 신뢰하는 부분이 확실히 다를 것 같고요. 미국 시장이 문제인데 미국 시장은 어느 정도의 가격 경쟁력을 분명히 유지를 해야 될 것 같고요. 저희가 브랜딩을 뭐 하긴 해야 되겠지만 그리고 거기에 안에서 이제 저희가 그 구조를 좀 배우는 시간은 필요할 것 같은데 다행히 저희가 이 합병을 통해 가지고 아까 말씀드렸듯이 가격에 대한 플렉서빌리티를 가져간다는 건 그런 경쟁에서 분명히 장점이 될 거라고 생각은 하고 있고요. 마지막으로 그 말씀하셨던 정책적인 부분은 아까 삼성바이오로직스도 뭐 충분히 지원을 받았다고 하는데 저희도 마찬가지인 것 같아요. 처음에 저희가 여기에 들어올 때 그 뭐 인천시에도 굉장히 적극적인 지원을 받았고, 그다음에 저희가 바이오시밀을 할 때도 뭐 많은 부분 세제적인 지원이나 이런 것들을 정책적으로 해줬기 때문에 그런 부분에서 좀 뭐 정부의 역할이 굉장히 컸다, 인천시의 역할이 굉장히 컸다고 생각을 하고 있고요. 다만 이제 신약을 할 때 이 안에 만들려고 하는 그 이 제조 중심의 인프라를 앵커 기업이 되면서 저희 같은 회사가 신약을 개발할 수 있는 앵커 기업이 되면서 거기에 지금 K바이오랩이나 이런 데 그 들어오는 기업들이 신약을 플랫폼이나 아니면 특정한 아까 말씀드렸던 그런 영역에서 개발할 수 있는 능력을 갖고 있다고 하면 사실 아까 말씀을 하실 때 이 값어치가 너무 이제 경쟁력이 없다고 하시는데 사실은 레키로나 개발할 때도 그렇고 제가 보기에는 저희 그 진펜트라도 그렇고 이걸 막상 개발해가지고 판매를 하고 잘 팔리면 그때 경쟁력이라고 표현할 수가 있는 거잖아요. 그러니까 이건 사실은 결과론적인 부분인 것 같고요. 사는 사람 입장에서는 자기가 마음에 들면 살 것 같아요. 그래서 그 경쟁력을 갖게 만드는 건 오히려 그 기업 안에 있는 것들을 어느 정도까지는 이렇게 보여줄 수 있는 단계까지 끌어올릴 수 있는 그 구조가 만들어진다고 하면 국내 그 뛰어난 인사이트를 갖고 있는 연구자들이 많기 때문에 분명히 그 선순환할 수 있는 그런 생태계가 만들어질 거라고 생각하고 있고요. 뭐 이미 제가 보기에는 신약이 나오고 있다는 거는 그런 생태계가 이제 어느 정도는 조금씩 시작이 됐다고 저는 이해하고 있습니다.
네, 감사합니다. 또 다른 질문 혹시 없으신가요? 네, 그럼 이것으로 세션 4를 마무리하겠습니다. 장시간 감사합니다.
감사합니다.
네, 수고하셨어요. 전화가 계속 와가지고.